Bismillahirrahmanirrahim. Ladies and gentlemen, thank you very much for joining us here on this SPF workshop on behalf of General Directorate of Fisheries, Ministry of Environment, Water and Agriculture, Saudi Aquaculture Society and National Center for Environmental Compliance. I would like to welcome you all here. We hope that this workshop will be a great learning opportunity regarding the SPF animals, productivity and market access. I am also very thankful to the presenters here. They are the industry experts. Hopefully they will be putting light and imparting knowledge to all of us. So with the name of Allah, I am going to start this workshop. First of all, I would like to request Dr. Victoria Lesans from National Aquaculture Group, having a vast experience regarding SPF animals, and she is the head of biosecurity, and she will be presenting regarding the SPF, SPT and SPR animals. Dr. Victoria, please.
And the confusion is also coming because of the interest of companies selling broodstock. In fish, we don't have this issue because there is very little trade on broodstock. So it's only in shrimp. So the thing is, the different commercial companies, they come up with SPF, SPR, and they promote this, they use this for commercial purposes, and they misuse them. So, also, there is a misconception, and depends very much on the continent. In Asia, they think SPF animals are more robust. They are stronger, they tolerate better diseases. But if you go to Latin America, they feel that SPF, they are very weak, that they get sick very easily. It's just because of the different experience of the industry. In Asia, CP, basically it was CP, imported SPF from Hawaii, and together with a biosecurity strategy, they had very successful results at the time of yellowhead and white spot. So they thought the SPF animals are stronger than the normal ones. But it's not, it's only because they combine it with biosecurity. In Latin America, they did the opposite. They brought SPF animals from Hawaii, they put it in ponds, no biosecurity, and everything died. So they thought, oh, SPF is very weak. So the same animals, they are perceived very differently in one continent or the other. Also, people think, oh, SPF grows much faster. Not necessary. SPF only means that they are clean. Or they say, oh, SPF, they have inbreeding. Not necessary. Depends on the breeding program. So this is very important to understand all these concepts because it has implications on how you manage the health of the animals and the risks that they involve. So trying to clarify these concepts, we got the support from FAO, and we got together, James Brock, he came to make an audit of our breeding program. He's one of the persons who developed the first SPF stocks in Hawaii. We got Tim Flegel, which is the person who also did an audit of our breeding program, and he's the most knowledgeable in terms of the relation of the shrimp and pathogen. Robbins Mackinson, who was the one to bring SPF to industrial scale, because before it was only used for experimental work. Melba is from FAO, Marcela is from Latin America, and Rohana is former FAO. So, just a bit of history. The first SPF stocks were developed in the US in the 80s, following the concept of poultry. Yeah. So what they did is they collect animals from the wild in areas that were far away from farming areas. So there were less chance of infection. So they went through quarantine and screening. So they went to look for animals they have not been exposed to pathogens to have more chances of finding the animals we were clean. So they were used for experimental work, for nutrition, and for genetic development. But it was only experimental work. So it was only Mackintosh in Belize, and then when he moved to Thailand, that he used these animals at commercial scale. So, and that is what allowed, this is the Thailand production when it was hit by white spot. They introduced the Vanamay in red. They were using monodon. They introduced the Vanamay, and that allowed the exponential growth. This is Ecuador. They were hit by white spot. They tried SPF, but no biosecurity, so everything died immediately. And it took them 8 years to recover production. So the definition of SPF, a specific pathogen-free. Sorry, I missed in the title. So it's a population that has been tested negative for specific pathogens for at least 2 consecutive years. It has been raised under high biosecurity facilities, under stringent biosecurity measures, and has been fed with biosecure feeds. So it requires a surveillance program in place, both at molecular and histopathological level. So even if the concept is clear, there are many gaps, because we say it has tested negative for specific pathogens. It doesn't mean tested for all pathogens. They may be SPF for white spot and IHHN. Okay, so it's only for specific pathogens. It doesn't mean they are free from all. So whenever you want to buy an SPF animal, you need to ask, what are they free from? Your list can be very long or can be very short. It can be the list of the OAE or it can be your own list. So you always need to find out. So, for instance, in NACWA, we test against all known pathogens, listed or not listed. We include all known pathogens. As soon as a new pathogen is listed, we include it in our surveillance program and we check for it. We say it has been raised under high biosecurity facilities. What is a high biosecurity facilities? It's not the same a facility that is in the coast, that is in the middle of the desert. You need different measures, no? So that is not defined. We say, well, stringent, yeah, biosecure feeds. That's another problem. There is still no maturation feeds which are biosecure. We still use fresh feeds. So that's another big gap. So we test the feeds, but those biosecure feeds still don't exist. Then we say it requires a surveillance program in place. Okay, but no one has decided how the frequency of sampling, the stage of sampling, the size of the sampling. So as you see, the concept of SPF is there. It means that we have a clean population, that we have proven it for at least 2 years, but it's not, you know, there is a lot of gaps. Yeah, no one has defined this. FAO wants to work on it so that there can be a certification program from SPF, but it's very dangerous because of the liability. So this is pending. So this is not defined yet. So there are 2 approaches to develop SPF. The original one that used in the US, so they look for wild animals in areas that were not affected by farming, so animals that were not exposed by pathogens. And the opposite approach, that's what we call the reverse SPF, and that's what we did. Instead of going to remote areas, we went to the farms where the viruses have been present for at least 10 years. So we searched for animals that were clean at a big size, so between 25 and 30 grams. So we thought that animals of that size in an area that is endemic and they are still PCR negative, it means they have some characteristics of tolerance or resistance. So these are the 2 approaches we use. This is the production in Saudi Arabia. First detection was 2010, 11 and 12. We tried the approaches that have been used in Asia and Latin America. None of them work. So we defined a new strategy. We switched to Vanamay. We use these SPF animals developed from endemic areas, and that allows us immediate recovery of production. Yeah? So these are 2 different approaches to SPF development. If you look at papers, you will see that the definition of resistance and tolerance depends on the background of the authors. For a geneticist, it only relates to the relation between the pathogen and the shrimp. if the one who writes the paper is a pathologist, you know that the disease, it always has three components. One is the shrimp, the pathogen, and the environment. Because we know that the environment is a cause of trigger of the disease. You can have, please come in. You can have the infection, but no disease, but then temperature drops and you have an outbreak, or temperature goes up and you have an outbreak, depending on the pathogen. So you will read papers by geneticists and they refer all the time to resistance, not tolerance. But that's because they only look animal pathogen. Yeah, so it's not correct if you are looking at it from the animal health management. So the definition of SPT, specific pathogen resistance. So these are the stocks that are refractory to infection, even if you challenge them. So even if you inject them with a virus, they will not get infected. So that is resistance. They don't have receptors. Yeah? So this resistance, it can be specific to a pathogen or a strain of pathogens or to several of them. So when you see in publication, they say, or advertising, our broodstock are SPR. SPR for what? Are they resistant to all pathogens? Not possible, because each of them has a specific mechanism. Yeah? So you always need to find out what is resistant against. Also, when you see the advertising, it says SPR, you don't know what is the health status. It might be tolerant or resistant to white spot, but it might be infected with EMS. We don't know. So we need them to be SPR and SPF, because the health status is defined by SPF. This is only the genetic response to a disease. The only population that is SPR, that is resistant to white spot, is a population of monodon that was developed by Grace Law, Professor Law in Taiwan. She was never able to bring it up to commercial scale because she wanted to be able to sell them. So we don't know exactly what happened. She found some animals from the wild that they were truly resistant. Even if she injected them with white spot, they wouldn't get infected. But the heritability of this resistance was very complex. So she was not able to come up with a commercial population. The other concept is a specific pathogen tolerant. And these are the shrimp in Saudi Arabia. These are stocks that are susceptible to infection. You remember, resistance, they cannot get infected. Tolerant, they get infected, yeah, to a specific pathogen, but they do not develop the disease, or they develop it to a lesser extent. So before with Endicos, white spot hit us and we had a wipeout. We had 99% mortality. Now we have animals which are tolerant, yeah, and mortality can be maybe 10, 15%, depends on the weather. If it's in winter, we'll have higher mortality, yeah. So again, it does not, SPT does not refer to the health status. It can be tolerant to white spot, but can be infected with EMS. So if you want to buy animals, you need to find out what is tolerant against, and then what is the health status. And most Latin American stocks can be considered SPT. They always talk about SPR for Latin America. They are not resistant, they get infected. In fact, right now, in Ecuador, there are high mortalities due to white spot because it's La Niña. It's an environmental, you know, El Niño, La Niña is an environmental process, and now temperatures are very low. So all the Taura area and coastal area, they are having mortalities due to white spot. So they can get infected. Normally, they will not show disease, but under certain circumstances, you can have mortality. Then we have another term that is used in the industry, APE, all pathogens exposed. In fact, instead of calling it APE, we should call them uncharacterized selected survivors, but no one is going to use this term. So APE, it says that these are animals that are collected from the ponds and they have been exposed to all pathogens. We cannot say all pathogens, but because there is no place where we have them all. Yeah, but this is a term that is used by the industry. Again, we don't know what is the health status, whether they are infected or they are not infected. We don't know what is the genetic response to pathogens. We don't know if they are susceptible, they are resistant, they are tolerant, we don't know. And they present a very high risk of movement and introduction of pathogens. Then we have the high health. That's another commercial name that is sold from Hawaii. And high health only refers to descendant of SPF stocks. Again, they are descendant of SPF stock, but we don't know what is the health status. Have they been kept in the same biosecure conditions? Are they still clean? Are they infected? And we don't know the response to the pathogens. Are they tolerant? Are they resistant? Are they susceptible? So these are terms that we have to be very careful because they are in the market and they are promoting this concept. So it's very important to understand them and always try to find out more. What do they mean? Yes? So understanding, what is the health status and what is the genetic response of disease allows you to define what is your biosecurity strategy. For instance, the animals, if we look at health status, they can be SPF, it means clean, or they can be APE, they can be exposed to pathogens. And then when we look at the genetic response, they can be susceptible, they can be resistant, or they can be tolerant. And then if we look at the productivity, SPF normally has a high growth because they have been in genetic programs that have improved the performance. Or they can have high salinity tolerance, for instance. So you are going to define what are your facilities and what kind of animals you want to use. If you have high biosecurity facilities, if you have an indoor facility where you can control the water, you can disinfect it, okay, then you go for high growth. And if the ones with the high growth are susceptible, you don't care as long as they are clean because they are not going to get infected in your facility. But if you have facilities like in NACWA, we have 10-hectare ponds, you cannot treat the water, you cannot prevent the access of crabs and so on, your priority is going to be to have animals which are tolerant or resistant. Yeah? So in addition of tolerance and resistance, you want to start with clean animals. Starting with clean animals, it helps you to ensure that you can produce consistently. So it all depends at the end. What are your facilities? And based on that, you decide what health status I want and what response to pathogens I want. So it's very important to remember, SPF only means that it's clean. But clean is not only a PCR negative. A PCR negative, I can give you infected animals are PCR negative. I can play with the sample size, with the number of cycles of the PCR. I can play with temperature. So the only way to ensure that your animals are clean is to have this continuous testing a minimum of 2 years. Yes.
Just one quick question. Being an SPF right here,
Yes.
so in one lot
Yes.
or in one stock,
Yes.
you may have susceptibles, SPRs and SPDs
Yes.
all in there.
Yes.
It can be.
It can be.
And they will function definitely, respond definitely differently to the bacteria.
We see that in our breeding program. We do challenge tests to see the survival against white spot of the different families. If we can see, some of them are more tolerant than others. Some of them we have had 100% survival, but we are not confident that they are resistant. Yeah, we don't believe, we always have to be on the worst case scenario to be on the safe side. But you can see that the tolerance is quantitative, resistance is qualitative. Either gets infected or it doesn't. While tolerance is quantitative, can be more or less. So we see, for instance, CP animals. When we do challenge, very high mortality, very little tolerance. They are all SPF, CP and NACWA, all SPF. But the response to pathogen is different. So CP, very high mortality, NACWA, very high survival. So it's the tolerance.
But you can pick and choose right in that time, at the time of their, at early stages.
Yes, we challenge in early larval stages, and whatever is below a certain survival level, that's discarded. This doesn't go to commercial purposes. Yeah, it's kept for the breeding program because there we are trying to maintain maximum diversity. Because maybe, next year there is a new pathogen and we need to select a new trait, so we don't know if that trait we can find it in that population. So we always try to maintain the widest genetic variety. Yes? Yeah, so you need to define the sanitary strategy, yeah? The objective is always to reduce the economic impact of the diseases so that you can have a consistent production, that you can predict your production. It cannot be that, everything is going well and then weather changes and you have lower temperatures and then you start with mortality. It's also important for transboundary movement of pathogens. If we start moving animals, we have a very nice history of white spot being moved from Asia to Latin America, Taura being moved from Latin America to Asia, IMMV from Brazil to Indonesia. So we have very nice history of movement of pathogens. So the use of SPF prevents this. So the strategy, you need to base it on the profitability. So it's your infrastructure, your investment, the technical level, what is the growth you expect. Yeah, so the recommendation is always to use SPF animals. Don't work with animals you don't know the health status, that can be infected. Because that means, you are invested in infected animals. It is playing a lottery, yeah? Animals can get infected when they get into the pond, but the transmission is very inefficient when you compare it with a transmission from broodstock. From broodstock, vertical transmission is very efficient. In NACWA, when we first detected white spot, we started detecting it in the ponds, and there was no mortality, very, very little. So we thought, our animals, our indicus is tolerant, is resistant. No. The moment it entered the breeding program, and we had vertical transmission, it spread the whole NACWA, and it was wiped out within 2-3 weeks. No, it was really, temperature went down and everything was gone. So always SPF. In shrimp we are very advanced. In fish we are not. We are still using wild broodstock. We are using cage broodstock, and by exposure to pathogens in the wild, because the pathogens, they are not created in the pond or in the cages. They are coming from the wild, reservoirs in the wild. And we know that in the Red Sea, we have VNN, we have iridovirus, we have plenty of things. So by using animals that have been in cages, they can be infected. They will not show the sign of disease because, for instance, VNN only causes mortality in the hatchery. Yeah? So we need to invest in development of SPF for fish. And I think that is all. Thank you very much.
Thank you.
Any more questions?
Thank you very much, Dr. Victoria, for the very informative presentation. NBC, the Nuclear Breeding Center, surely the backbone for the production of SPF animals, and surely the biosecurity of NBC is a priority. So regarding the biosecurity, surveillance, and related protocols, I would like to invite Mr. Peter Paul Rivers for the next presentation. Sorry, gentlemen, our next presenter is Mr. Peter Paul Rivers, and he is presenting regarding aquatic disease diagnostics. So, Mr. Paul, please.
So once again, good morning, everyone. I'll be presenting to you the aquatic disease diagnostics. So I'll try to simplify some terms so that each one of us can relate, because it's kind of technical and complicated if we will do each of the processes that we do during the diagnostics. So as a review, so the definition of a disease is it's a deviation from normal condition, either behavioral, structural, or physiological state. Deviation meaning you have this normal condition, you observe your fish, your animal, for example, and if there's a change in their behavior, for example, their swimming pattern, their movements, or there's a change or a shift in their structural. When you say structural, it's more of a tissue or cellular level. And physiological, it's more on physical, their growth, for example. So if there's a change, there's a deviation, or there's a shift, so we can say that maybe we can assume that your animals has a disease. Okay. Then, what are diagnostic tests? So these tests are a series of tests designed to determine the health status of a healthy population of animals versus that of a population of animals deemed unhealthy. So we check our animals using different diagnostic tests or techniques to check if it's either healthy or sick. Okay. And the aim of disease diagnosis is to determine the accurate cause of the disease, then select the proper treatment. Okay. So for example, that's just a cartoon picture of a pathogen. So a pathogen is anything. It's either a microorganism that can cause the disease, or it can infect the host or the animal. So a pathogen can either be a virus, a bacteria, a fungus, or a parasite. Okay. Then after you check for the cause of the disease, then you can recommend the treatment after. So that's disease diagnosis. Okay. So what should be included in the diagnostic evaluation? So for example, there's a case of mortality in a pond or in a tank. So we should check first the case history. So what is the case history? Those are the previous information that we can get. For example, for the animal, we can check for the mortality rates, how many mortalities these past few days or past few weeks, if it's increasing or not. Okay. Or the mortality, is it sudden? Okay. And those are some characteristics of the animals. What are the species of the animals? What's the size? It's either a larvae or broodstocks. So all information that we get from the animals, we should take note of that. And then another one is the facility or husbandry. It's more of a management aspect. So is there a sudden change in the routine management? And also for the feeding, is there a change in the diet recently introduced? Or change in quality or forms of your feeds that you used? Or did you use any prior treatments, like some medications? Okay. Then the other one is the environment. So aside from the animal, the husbandry, the last is the environment. You can check for the stocking rate, for the water, if there are presence of natural enemies, or those are, for example, some birds. There are bird activities, there are crabs, or there are other species aside from the one that you cultured. Or are there any news or information that maybe there are infected animals from the adjacent or the near neighboring farms? Then aside from that, we also include the water. So how often do you exchange water? And what are the physical and the chemical characteristics of your water? So for physical, you can include what's the color of the water, the transparency, or you can see some algae in the water. And for chemical, you can also check for the dissolved oxygen, the ammonia, the pH, the nitrite. Those are the examinations of water, if there are some changes from the normal range. So all of these changes, all of this information, we need that one to fill in a submission form so that we can correlate our results later on with the case history. So also here you can put some description and some parameters in water and others, all that you can provide from these three aspects. So the purpose of the sample submission form is mainly to collect information. So on the clinical presentation, also it is to dictate the documentation needed to demonstrate the traceability of the sample is maintained throughout the receipt, testing, and the storage process. Okay. So before you submit sample, you should fill all necessary information. So here, I make I put three different levels of diagnostics and their associated requirements. So level 1, level 2, level 3. So usually for level 1, those are from the pond or from the facility. Okay? Those observations of animals and the environment. So it also includes gross clinical examination. So usually it's on the field. So the people involved there are maybe farm managers, farm technicians, some aquaculturists, or some biosecurity officers also. Okay? So their work requirements include they should have knowledge of normal feeding, behavior, or growth of stock. And also they should regularly observe the animals in the tanks or in the ponds. And they should have a good record keeping, including fundamental environmental information. And they should know how to preserve and collect samples to submit for the lab diagnosis. So the level 1, it's mostly on the field, and they should have contacts for level 2. So for level 2, we enumerate here mostly of the lab analysis. So for level 2, it's more on laboratories. We have the parasitology, you have parasitologists, bacteriology, mycology. Mycology is for fungi, and histopathology. So this belongs to level 2. So for level 2, these are laboratories with basic equipment and personnel trained or experienced in aquatic animal pathology. So they require moderate capital and training investment. Moderate capital because you need to invest on some machines, equipment, and you have to hire or train operators of that machine. Okay, so they should keep and maintain accurate diagnostic and laboratory case records. Then the third level includes virology, electron microscopy, molecular biology, and immunology. So these are highly equipped laboratory with highly specialized and trained personnel. And this comprises the types of advanced diagnostic specialization which requires significant capital and training investment. So this is good if you want to do more research aside from diagnosis. Okay. And what are the basic steps of diagnostics? So here is the flow diagram of what we are doing for the diagnosis. First, you have the sample. There's the aquatic animal, for example, the fish, and then you check for the gross clinical signs of the disease. Then you collect the sample and submit to the lab. So in the lab, you have different techniques to use. For example, microscopy, histological, microbiological, immunological, and molecular techniques. So in the next slides, I will show you one example of each of these techniques. Okay. So clinical signs and gross lesions. So what's the difference between symptoms and clinical signs? For example, for humans, you talk about what you feel, for example, to a health specialist or to a doctor. That I feel some pain, body pains, or headaches, or something like that. So that's symptoms. But for fish and shrimp, for example, they cannot say something that they feel pain here or somewhere in their parts. So instead, we use clinical signs. So clinical signs is more on observation of the physical condition or behavior of the animal. So symptoms, it's more of something that a description of feeling. So we cannot ask them and they cannot tell us. So we are the second person to observe these signs, okay, so that we can check for diagnosis. For lesions, we call lesions all the abnormalities that we can see in the shrimp, especially on the physical. For example, there's some wounds or some abscess or some polyps or some tumors in the body of the animal. We call it lesions. It's gross because it's not good to look at. Okay. So these are some examples for the clinical signs and gross lesions for shrimp. Okay. So we have moribund. When we say moribund, it's weak. It's about to die. So it has weak behavior, black gills. So this is an example of a black gills or brown gills. Then melanization or dark spots in the exoskeleton, this one. Then dark or red discoloration, for example, and soft shell. So there are more other lesions and clinical signs. So I just selected some. And then for fish, these are the clinical signs or gross lesions for fish. For example, there's abnormal skin coloration, irregular breathing, there's abnormal swimming, there's reduced feed intake, reduced growth rate, there's skin damage here, and runt or skinny, or it has a bulging eyes. So I think it's exophthalmic, and this is hemorrhagic. So those are examples of lesions for fish. Okay. So do clinical signs and lesions allow you to make diagnosis? So if you already see some lesions or some clinical signs in your animals, can you diagnose already? Not yet. Because clinical signs and lesions suggest to you the possible disease process. Okay? So that diagnosis can only be made by confirming the pathognomic lesions caused by or isolating or demonstrating the presence of pathogenic organism with a concurrent disease process. So we need to know the pathogens and the lesions caused by that pathogens. Okay? So how many samples do I need to take for diagnosis? So I listed here 2 sampling procedures. First is the random sampling, and the other one is the target sampling. For random sampling, this is non-selective. Okay? So this is good for population testing to get the idea of a disease. So for example, you see this table. Okay? You want to take representative samples from a pond or a tank. So this table suggests to you the percent prevalence and how many representative animals you will take from the pond. Okay? So prevalence, it's usually expressed in percent. So it's a percent on how much or how many number of diseased animals in a total population a certain period of time. So if you want to know the status of the whole pond or of the whole tank, you should do random sampling. For example, you have more than 1 million animals in a pond. At 2% prevalence, you'll take around 150 samples. So it's a random. You can choose, for example, 4 different sites. You do cast nets or you do bucket. Then you count and you check how many have this kind of common characteristics of disease, for example. So for target sampling is for the disease diagnosis. It's a careful selection based on clinical signs of animals for disease diagnosis. So for target sampling, it's good for checking if there's pathogen or not. So usually you should visit, for example, if you visit a pond and you can see some weak animals swimming near the inlet or the outlet of the pond, or you can see some moribunds here, just collect those animals, maybe around 5 to 10 pieces only, then you submit to the lab, because those have these clinical signs. Okay, so target sampling is essential because, for example, here, if you do random, you cannot easily detect the pathogen here. Okay, but if you already know that these are the clinical signs of your sample for the disease, then you can get the correct sample, and this will give you correct information. It can give you correct analysis results, and it will give you correct decision also. Okay. So that's why target sampling is also important here. So let's proceed to the different techniques for diagnosis. First is the general necropsy, wet mounts, organ imprinting. So these are microscopy techniques. So you need a good microscope for this one. So this technique, for example, your specimen is placed between the slide and a cover slip. Okay. If it's wet mount, you can put some saline water and check on the microscope. If it's tissue imprints, you can put some dye like crystal violet and view in the microscope. So the objective is to determine the gross signs of illness that caused the death. You can check for the external and internal gross examination. You can do wet mounts for gills and skin scrap. You can do tissue imprints of spleen, lower gut, lesions of any. You can also do gut content analysis. You can do blood smear, and you can collect sample for other analysis like bacteriology, histopathology, and virology. So usually, it will just take 1 hour. For example, one person will do the wet mounts, one person will do the tissue imprints, and one person will do the gut content. It will finish fast. So it's kind of rapid test also. So for the pros means the strength of this technique, or the advantage is that this is inexpensive, so it's cheaper. You need only a microscope and some small reagents. This is suitable for resource-limited setting. It's a rapid detection of certain parasites and bacteria. So rapid meaning it's fast. You can get the result right away within the day or within hours only. And this is good for checking parasites and bacteria. And the disadvantage of this, it can be labor intensive. Okay? So you need also skilled personnel. And it's not always possible to identify the agent because it's limited only to certain pathogens. Okay. So these are some of the pictures of a necropsy and wet mount. So you have these basic tools that you use. Then for fish necropsy, you can dissect the fish, the external and the internal organs. And also we have these skin lesions here, these exophthalmic eyes, these bulge eyes. And you can also see during necropsy some parasites. Okay? And these are hemorrhagic eye, gill, liver. You can take samples here and put in a slide and check in the microscope. For example, here you can see some round-shaped bacteria for Streptococcus. Okay. The next technique is histopathology. Histopathology, it determines the changes that occur in the tissue due to pathogens or disease. Okay. So you can see the presence of parasites, bacteria, viruses, fungus, as well as lesions in tissues caused by their propagations, lesions related to nutritional deficiencies also. So there's a lot of procedure for this one. That's why it takes 5 days. So it's not rapid. You have to wait for how many days to get the result. So the procedures include fixation, decalcification, dissection, tissue dehydration, clearing, infiltration, embedding into paraffin blocks, cutting paraffin blocks and mounting, sections and glass slides, staining, and check for the microscopy. Okay. So the advantage of this is you can do presumptive diagnosis. It allows an evaluation of infection distribution in various tissues, useful in assisting diagnosis of disease, particularly where the causative agents of new disease have not yet been identified. So the disadvantage is it might not detect low level of infection, and it's time consuming, and it depends on the expertise of the pathologist. Okay, so these are some of the pictures for histopathology. You have this shrimp, you fixed it. Then this is the cross section of this cephalothorax, and you put in the tissue cassettes, and this is the automatic tissue processor. Okay, you embed it with the wax. So this is an example of a shrimp embedded block, and this is for the fish. Then you do sectioning using microtome, staining, then check in the microscope. This is just an example of a tetrahedral baculovirus that you can see. Okay, so it's more of a lesions. You cannot actually see the virus there. So it's just the lesions caused by this baculovirus. Okay. Next is bacteriology. It's a microbiological technique. It is used for the identification, classification, and characterization of bacterial species. So the samples were analyzed through the growth of suspected pathogens. Okay. Then after you grow the pathogen, you isolate the colonies and identify using advanced biochemical identification system. So first, you do sample preparation. You streak it on a media or plate. Then you incubate it for example, 12 to 24 hours. If there's some bacterial growth on agar, you test for biochemicals. That's part of the identification. And we have this automated detection system that you just put there and it will give you a result. So you can finish this in at least 3 days also. So it's not rapid. You have to wait for 3 days because you need to grow. So for the advantage, it's useful because the pathogen is isolated and the causative agent can be confirmed. So bacteriology is useful for treatment validation. So for example, in tanks, we do swabs to check if there is some presence of bacteria before putting water and animals there. We can check also bacteria in water so that we are sure that the animals are clean, the water is clean. So the disadvantage is that it's labor intensive, it can be expensive, and it's not always possible to confirm the identity of the causative agent. Okay. So these are some of the photos. For example, this is your tissue. You take swabs here for the internal organs, for example. You streak in a selective or differential media. Then you incubate for 1 day or 2. Then if there's some growth, okay, this is an example of some bacterial colonies. This is Vibrio species. Okay, you can also do susceptibility test to antimicrobial. Okay, and you can also check for gram-positive or gram-negative staining. So I think most of the fish bacteria are gram-negative. And this is the automated VTech2 compact. So you just have to load your colonies here with some reagents and it will give you the name of the bacteria. Okay, so, and these are the different test cards that you used here. So in the result, it will show you how much % probability that will match on what is that species of bacteria present. Okay. And next is ELISA, or that's enzyme-linked immunosorbent assay. So it's an immunological technique. So this one, it's for the detection of antigen or antibody. Yes?
Sorry, I have questions. How do you decide what kind of test you do? Base it on the morphology, tissues, or what exactly?
Yes.
When you decided what kind of test you
Yeah.
PCR or histopathology, base it in what? Is it in the shape or is it in what?
Usually, we check first the clinical, okay, the clinical signs, because clinical signs, there are already some literature that associates to certain pathogen. Okay, for example, for shrimp, if the rostrum is bent, so most probably it's a virus, IHHNV. So we need to do PCR for that. Okay, So if there are some skin lesions, for example, because if you have wounds, most probably bacteria will attract that one. So you can do preliminary tests like general necropsy, microscopy, check. Then you do bacteriology also. And if you have specific kits for that bacteria, you can do PCR also for that one. So as much as possible, we try to do at least 2 techniques to confirm that one. Microscopy are just preliminary, also bacteriology. We need to do PCR also for confirmatory.
Based on the symptoms that
Clinical signs also.
clinical signs.
And the history of diseases in your facility. That will give you a lot of.
If you have a history of cases of white spots, so all the samples should analyze also with white spot or some bacteria in cages or in monks. Okay. So for this ELISA, it's more of a detection of antigen and antibody. So antigen is anything that is foreign, okay, that comes inside the body of the animal. So antibody, it's a protein or immunoglobulin that is automatically produced by the body to fight this antigen. So we can use this ELISA for the detection and quantification of pathogens. And we can also use this one for the detection of antibody present in the vaccinated fish. So the procedure is this. It will take 4 days also. Okay. And the advantage of ELISA is that it's a versatile method to identify pathogen. So it can identify if there's a bacteria or something. And then the antibodies present. It's you can know if it's the immune system of the fish is okay or not to fight these pathogens. And it's quantitative. It's sensitive. When we say sensitive, it's the ability of this technique to test. It can detect low-level infection. It's non-lethal, meaning you don't need to kill the animal to take samples. You just need maybe 1 to 2 milliliter of blood for this one. And the disadvantage of this is standardized reagents and specialized equipment needed. So you need careful selection of controls and a skilled operator. And it's time consuming because it takes 4 days. Okay. So these are ELISA photos. You collect blood from the fish, for example, and you take serum from this one. And then you have this culture to test for that one. You have the optical density. You have these plates. It's more of a color changes. Okay. And then you read it in the plate reader and you get these results. Then from that you can interpret whether how much is the antibodies produced by the fish. Okay. And the last technique is PCR. So it's a common. It's polymerase chain reaction. It's a molecular technique. Okay. So the principle is the detection or attempt PCR with primers that are complementary to part of the genome of the pathogen. So it's ideal for detecting the presence of bacteria, viral, parasitic, fungal pathogen. So it includes nucleic acid extraction, master mix preparation, replication using thermal cycler, and you check for the CT number or the threshold cycle number or the DNA copies in the real-time PCR machine. And it takes around maybe 4 to 6 hours. So it's a rapid detection. You can get the result within the day. Okay. So the advantage of PCR is that it's a rapid technique and increased sensitivity and specificity. So it can detect low-level infection. The disadvantage is it requires well-equipped laboratory structure and well-trained personnel. It's more expensive, and then contamination can lead to false positive. So you should be very careful when doing PCR. Okay. And these are some of the photos. You collect tissue, for example, and we have this automatic nucleic acid extractor. Okay. You can check the concentration of your DNA or RNA using this nano drop, and you do master mix preparation. And for PCR, it's a specific kit. You have specific kit for specific pathogen. Okay. And then these are conventional method. You do electrophoresis, for example, check in the gel duct, and you get these bands here. Okay. If there's a band here and band for positive, that's positive. It's more of a qualitative. And if you do real time, so you'll get some amplification bands here, and you'll get the values for how many copies of the DNA of the pathogen present. So if it's less than 100 or 1,000. So these are other advanced diagnostic techniques, examples of novel technologies with potential use in aquaculture. You have this DNA fingerprinting for genetic sequence, microarray, okay, for gene sequencing. Okay, you have also this. You can also do this in situ hybridization. You can buy electron microscope also. Okay, and there's some immunofluorescence staining. Okay. So what should the lab tell you? So, for example, Tinacibaculum was isolated from the skin lesions of deceased sea bass. So the causative agent, it's a causative agent of Tinacibaculosis. Okay. So what is the final outcome of the diagnostics? Is it morphological diagnosis only? For example, in wet mouse, you see it's long, thin, rod-shaped bacteria. When you do bacteriology, you can see that it's pale yellow colonies and even edges. Okay. When you do histopathology, it shows Tinacibaculum-like bacteria. Okay. And when you do PCR, for example, it's positive, severe positive. And we do genomic characterization, it shows that it's a strain of T. maritimum. Okay. Then what recommendations regarding husbandry? So you can either manipulate temperature and salinity to reduce morbidity, or you can control the fish density, you avoid overcrowding, you reduce stress conditions, and avoid overfeeding. So for treatment, you can do chemotherapy or give antibiotics, oral medication like oxytetracycline. Okay. You can do also prophylactic. Those are alternative to drugs. For example, you do routine formalin bath, you use potassium permanganate for water disinfection, you use hydrogen peroxide to treat the water and culture. And you can do vaccination also. Okay. And for the facilities or the environment, you ensure that the water quality is good. You have adequate dissolved oxygen, and you can keep the pond or the cage healthy. You disinfect tanks, the ponds, equipments, etc. Okay. And for the conclusion, so diseases are unavoidable business risk. Okay. And there are emerging diseases also. So target sampling is important for diagnosis. And a combination of methods is often required for a definitive diagnosis of disease. Surveillance and animal health management help to anticipate and mitigate potential problems. Both tools combined give us opportunities for early detection of diseases. And lastly, investing in biosecurity tools is insignificant when the profitability increases. That's all. Question?
Diseases are unavoidable business risk, as the conclusion shows here. With the increased aquaculture production, there is an increased trade between the countries, and it is equally increasing the chances of exotic pathogens to be introduced into other countries' aquaculture industry. So based on our surveillance program, we have faced this issue before, and still on everyday basis we are checking it, and we are finding exotic pathogens in imported consignments. So our next presenter, Dr. Chakravarti, is going to present the molecular diagnosis of exotic pathogens. Dr. Chakravarti, please.
Good morning to all. I hope already we feel sleeping mode. Again we are going to. Okay, I am going to present a topic on molecular diagnosis of exotic pathogens in imported shipment sample. Actually, the transboundary animals, especially the live animals which are brought to country, it's high risk in the disease when you the concern for the disease when you import or export the live animals. So that has to be checked and that has to be tested in a proper manner. So I'm going to discuss about little bit introduction about molecular diagnosis and what is the exotic pathogens. And we got a great experience on one of the exciting pathogen which was found in imported animals. So what is molecular diagnosis? Actually, molecular diagnosis completely deals with genomes and proteins. So study of DNA, RNA and proteins, which is called genomes and proteins. The entire set of DNA which is contains in the cell, which is called genome. So which might be an essential or non-essential, which is called in scientifically introns or exons. And proteins, entire protein, a set of proteins which can be expressed in the form of expressed. So the detections and analysis can be done with the genes and proteins. Here, for example, DNA, RNA and protein. And molecules of life, being it DNA or RNA, is it okay? Okay. Being it DNA or RNA or proteins, it's all have connections when pathogen meets with the host. So, for example, like pathogen when meets with the host, the DNA, the product of DNA, which means the essential part of DNA, particularly which is called genes, which can be expressed by the form of proteins that can infect to the, to the host. So, which is said to be a protein machines and central dogma of life. So, with this DNA and RNA, the molecular diagnosis can be done with the protein. So, with this, we can able to do the disease diagnosis, and the disease diagnosis can be done and which can be helpful for to monitor the disease and to do the aquaculture biosecurity. And with this all the techniques, and with these all the techniques with the molecular diagnosis, we can do the estimate the risk. For example, whether we need to go for the harvesting or partial harvesting. So then it can be helpful for the quarantine measurement. Those animals are imported, are going to be exported, or imported animals which needs to be kept in quarantine. Quarantine animals can be tested. And molecular techniques, there are many techniques available as of now. So I just listed some of the techniques which was utilized in aquaculture industry. For example, like PCR, real-time PCR and multiplex PCR. It's widely used in many of the aquatic industries, aquatic labs. And DNA microarray, NGS, next generation sequencing, Sango sequencing technology, and then mass spectrometry. Actually, this mass spectrometry being used in research labs, still now in diagnostic industry, we didn't get it as far as disease diagnosis. So because of the technique, technique which is need long process, and then RAPD PCR, rapid amplification of polymorphic DNA loci, will be used highly for bacterial genetic identification, and then in situ hybridization. And recent days, past decades, been used to analyze post histology analysis, and LAM PCR, which is being used mostly for high GC content, which is called example like Vibrio species, and like bacterial species can be identified easily. Next, and what is exotic pathogens? Already I said exotic pathogens which comes from other country to another country, which will be imported into your country, while imported animals which will brings to our home country. So which might not present in our country. For example, like those 2 years we were doing the detections, analysis, we might have any disease. So these minimum 2 years we shouldn't have any in our regions. These all called exotic pathogens. So these exotic pathogens must be listed or must be listed in our government eradication program, and it should be a noticeable disease. And these all, some of the exotic pathogens, for example, like bacteria and viruses and parasites. So with the molecular diagnosis, actually one of the interesting bacteria which was found Vibrio parahemolyticus. So actually initially with the microbiological technician, so people found that bacteria which might cause the infections, but all the bacteria which may not cause the diseases. So with the molecular diagnosis, with the molecular techniques by sequencing, they found one of the toxin which produce a bacteria and which cause the diseases. And again for the parasite, the best example now we are doing EHP, enterocytozoan hepatopeni, which produce the spores, and the spore, there is a gene called spore wall protein gene. So by this molecular diagnosis, so the spore wall protein genes can be identified. And next one. And what are all major economic important diseases? For example, like these all DNA diseases, DNA viruses and bacteria like WSSV, IHNV, DIV, BP, MBV, NHP, AHPND and EHP. And these are all for RNA pathogens like Thaura syndrome virus, IMNV, yellowhide virus, PVNV, pinase venomy necrosis virus, and CMNV. And some of the fish virus and bacterial diseases like VNN, RSI viridovirus, TALV, Streptococcus aureus, and Agalactica. There are only few I have listed. There are still many in the list, more to come in future. For example, recently for 2 months before, we have, China found picornovirus, pinase venomy. So it may listed in future more. And as per Saudi Arabia conditions, Saudi Arabia in the region, our Red Sea region, most of them pathogens are exotic. We did not have most of them. Only few of like Streptococcus or WSSV. So these are all we have found from our facilities. Okay. So one of the interesting pathogen I said, so the iridovirus family. So we found one of the samples which were imported from outside country. Okay. So iridovirus, actually it has a 6 genera, 5+1. Recently one classified which has a 6 genera, and iridovirus, chloriridovirus, lymphocystivirus, RNA virus, decapod iridovirus, and megalocytivirus. Decapod iridovirus, recently classified virus which infects the shrimps, okay, which is called decapod iridescent virus. So this one devastated in China aquaculture industry, especially like shrimp industries. Initially they found within a shrimp, then slowly when it has spread to entire decapod family like crabs, so then they found, they decided to name it as a decapod iridescent virus. So megalocytivirus infects the fish. Clinical symptoms includes, it shows lethargy, reduced appetite, severe anemic, and petechiae of gills. You will have a gills slight wounds and enlarged abdomens. And again, this megalocytivirus have 3 major genotypes. So one is Red Sea bream iridovirus, which was isolated or which was found from Red Sea bream from Japan, and infectious spleen kidney necrosis virus, so which was found isolated from mandarin fish from China, and turbot reddish body iridovirus, which was found in turbot fish from again China, and megalocytivirus is one of the which causes severe mortality, and it is a reportable disease to World Organization for Animal Health. So the sample, how we got it, the sample preparation, like it's a working procedure, like the imported samples collected from the JFRC quarantine, grouper fish fries, and around 48 animals we got it, including some of the dead pieces, and we made it into a pool. Actually we do it in a pool, each 10 fries, 10 per pool, and we have taken the tissues of brain, kidney and spleen. And the procedures what we followed, like extraction of nucleic acids from taco kit, and real-time PCR we used for IQ real kits for VNN, iridovirus, Streptococcus aureus and Agalactica, and conventional PCR. Followed by this, and conventional PCR we followed with procedure as well as to identify the species level. We followed the Kruta and Nakajima 2012 primers, and we did the gel elution and PCR product purification with Norgen. So again, for the sequencing, the PCR purified product, for the sequencing, we just send the PCR purified products to Malaysia, Apical Scientific. And bioinformatics work I did for with the sequence, raw sequence with the SmartSeq analysis, and raw sequences edited with the BioEdit program, which is available in freely in free software. And the NCB blast, after that by edited sequence, we made it to compare the sequence in NCBI. And MEHA10 and Clustal Omega just followed for the to find out the relationship among the genotypes. Okay. So next, again the primers used for the test for doing the sequence, RSIV and ISKNV genotypes to detect, which is available from OA procedure. So this primer is basically designed from the PST1 fragment, which is from lamellin-like gene, and for the sequencing, again for the to find out the megalocytovirus species, we just did with the major capsid protein, which is one of the important gene for construction and stability of the virus. Okay. So there is a results. Actually, when we did with the with the IQ real kit for VNN, Irido, Streptococcus inii and Agalacticae, and we found the positive for iridovirus, and VNN Streptococcus species it got negative, and the values really we got it like very high values, which means CT. So it is around like 21 plus or minus 1. So we wanted to do further related with this work. So we just go again, and the report was done, and we have given to the report to the ministry, and they have decided. Okay, based on our results, they decided, and they estimated the risk, and they did for the next level of work. As far as the lab work, again we did for same DNA, and we got from the WAP procedure, we got exactly around 570 base pair with a positive control and template control, which is previously tested negative samples, sample DNA, and negative template control, which is a water, nucleus free water. So we found that there is a something might be RSIV or ISKNV by doing the PCR. And for MCP major capsid protein, again we got around 777 base pair, and these products eluted and product was purified, and the purified product was sequenced, for example like WAP procedure with the lamellin products. So we got around 500 base pair. So this one had a great similarity with the E value of 0, which has a lamellin type ISKNV genotype. We did with the PCR with this primer, which can able to detect both. So with the PCR results, we can able to do, we can able to give the results, or we can, for example, decide the results only for might be which is an RSIV, but we doesn't know which kind of genotype. By doing the sequence and comparing the results in blast analysis, we came to know that so the iridovirus, it belongs to ISKNV genotype. Then by doing this with major capsid protein, again we got a greater identity with the E value, expected value of 0 with the ISKNV genotype. So we go for another, like I did with the phylogenetic relationship with this lamellin like ORF23 gene sequence. So which has, this is our sequence, which has a greater identity with the infectious spleen kidney necrosis virus isolates. So again for MCP sequence, these MCP sequence all which is linked or which had a monophyletic clade with all other MCP belongs to ISKNV genotypes. These sequences, which is called signature sequences, which made for the which is utilized for the utilized for the capsid, which is an which is used for the virus to make the stability. And then for the phylogenetic relationship, inferred with the whole genome with ISKNV with and another RSIV. So this is our sequence. We found that there are many ISKNV genomes of MCP it found in a monophyletic clades, and there will be a diphyletic. So because of this MCP sequence, what we got it's a partial. It's not an a whole gene identified. And there is a one tiger frog virus which found an extremely outer group, and this TRBIV virus which is found, TRBIV virus ISKNV, which is found in a diphyletic clade and extremely the outer clade. So which is especially when we said earlier, for example, like 3 major genotypes belongs to that. So we came through that. So out of in these 3 we found that ISKNV genotype which was found from our study. Sorry, again. Yes. Okay, I understand. Lower one. Okay. Yes. Okay. So the sequences we have submitted GenBank and NCB in the NCB, and it was published in these sequences and which is available and freely. You can get by these numbers. And with the conclusions, so molecular techniques, especially like PCR and real-time PCR, which is rapid, reliable and highly sensitive and which has a high specificity and accuracy. With the sequencing, so we can able to do the genotypic identifications. So for example, like one of the example, we found iridovirus with the PCR and real-time PCR. By sequencing, we found the genotype like ISKNV genotype, along with like WAP procedures and MCP identification. And of course, the imported animals, live animals, to be tested in quarantine facilities. It has to be done. So whatever the imported animal, especially we are bringing a lot of fry and eggs samples from, the animals, live animals from the outside country. So it has to be tested. And also after this or before this detections, the surveillance studies, KSA did not resulted any positive for iridovirus, any of samples, like more than 6,000, 7,000 samples we tested. No samples have got positive for those who are, the samples came from the facilities. So that is one. Again, the molecular diagnosis, again we need a more highly technical oriented people. And is it cost effective? It's a big question mark, because so high reliable test or high reliable results if we need to use high reliable kits. So the kits cost and the person who is going to work the technicality is all will have a high question mark as far as a normal like a form based labs. So only the labs, those can do some of the research, or in university labs, those have a budget, a business, budget for the research, they can able to do the things. Thank you. Any questions? Yes, ma'am.
What happened with that importation when you communicated that it was iridovirus?
It was actually it was in 2020 end 2021. It was just they brought around 500 pieces for the study purpose. So around early morning 3 o'clock from airport They got it. Morning 7:30 we got the sample. We got the results 3:30, and 5:30 biosecurity people finished up everything, their job. 5:30 means, I mean, we detected and we sent the report, and we made a decision that we need to kill the animals and all the things, and biosecurity people have done the job. That's it. So these all in the these all in the these things. Yeah.
Where from? Country?
Yeah, it is from I think Philippines. Philippines. Yeah.
Any other questions, please? No? Thank you very much, Dr.
Last time, still many in Eastern world compete on some disease. What is it?
There are many. I mean, there are like now picornavirus, kinase picornavirus. 2 months ago, it was in Chinese department, aquaculture department. They found it, so it might come based on actually the list economically important diseases are based on the survival or based on the mortality. For example, like here we can calculate like 40%. So if goes above anything 40%, there is some list and criteria they will follow. So they just generally they will pick 40%. So if goes above 40%, they will list it. So like what I said, coronavirus or white faces disease. There are many limb-sing diseases. There are many things are coming up now. So maybe in future it will come. Anything else?
Thank you very much. Although it was a very technical presentation, but I hope it will. It has shown to all of us that KSA has good capability to detect exotic pathogens. Okay. As I spoke earlier, the NBC is the backbone for production of SPF animals, and our next presenter, Mr. Madhu Abu, is going to present regarding the biosecurity protocols and surveillance at NBC. Mr. Madhu Abu, please.
Good morning, everyone. My name is Madhu Abu Chokra, Manager Broodstock Operations in Nucleus Breeding Center, Nakwa. Today I am going to present biosecurity protocols and surveillance program in Nucleus Breeding Center. Nakwa NBC is located in the Red Sea coast of Halith. It's around 180 kilometers from south of Jeddah. It is built in the island and isolated from shrimp commercial farms. This is our aerial view of Nucleus Breeding Center. This area-wise is around 160,000 square meter. All the boundaries are completely fenced, and all the production performed in only indoor buildings. There is no outdoor. And we have independent quarantine facility besides NBC around 300 meters distance. And also we have independent water treatment system where we are directly pumping water from Red Sea, and all the staff accommodations including management on site, and we have total 60 staff. What is Nucleus Breeding Center? Nucleus Breeding Center means a facility where specific pathogen free, already Dr. Victoria mentioned about all the SPF facility. SPF broodstock are raised over a number of generations in a highly biosecure environment, excluding the known pathogens by validating the stock's health and fresh feeds by surveillance program. Inside Nakwa, we have different compartments to mitigate the biosecurity risk. Compartment 1 is considered as high risk area. Compartment 2, highlighted in yellow, is moderate risk area, and compartment 3 is called low risk area. In high risk area, all the island facilities like Nucleus Breeding Center, quarantine facility, and biosecurity challenge facility, Nauplay production unit, larval rearing 1, larval rearing 2, larval rearing 3, larval rearing 4, and amberjack broodstock facility comes under high risk area. And in moderate risk area, we have phase 1 farms, phase 2 farms, and offshore cages and pre-shipment cages. And low risk area, all the township buildings and private accommodations, at work camp, and phase 1, phase 2 accommodations, and farm 108 processing plant and main processing plant. To enter island, Biocity online gate pass is required for all the vehicles, and also all the vehicles undergo vehicle disinfection at the entrance of the island gate. NBC staff and visitor guidelines. To enter NBC, all the visitors required Biocity Director approval, in this case Dr. Victoria. All the vehicles are not allowed inside the facility. And staff and visitors are required to sign in the log sheet. And shower and use of uniforms is mandatory to enter this facility. And foot and hand dip is mandatory to enter inside the production facility. Internal biosecurity. All the boundaries are with crab barrier to control the crabs, because we are very near to the sea. And also all the building equipments are equipped with anti-doors and signboards and insect traps. This is the layout NBC. In NBC production, we have divided into different compartments. In terms of staff and management, we have hatchery and breeding program wing. In wing A, wing B represents hatchery. In hatchery, we have larval units, maturation units, and live feed section. And wing C and wing G represents breeding program wings. In wing C, we keep all the brood stock for breeding program purpose. And wing G, this newly constructed wing where we keep individual families. We have a total of 85 tanks. We rear from TL to broodstock size. And next compartment is commercial multiplier wings and commercial multiplier raceways. These wings we are keeping all the broodstock for commercial purpose. In commercial multiplier wing we can keep around 100,000 broodstock, and in our raceways we are keeping around 200,000 broodstock. And then the last compartment is water treatment and reservoirs. We are managing with the different staff. And we have different sections, we have different uniforms. Hatchery team, commercial multiplier, and life support, we have different uniforms. And for the external maintenance teams, we are providing with blue color overalls. And for the visitors, we are providing with white color uniforms. And visitor movements inside NBC, all the visitors need to follow the biosecurity flow from low risk area to high risk area. In this case, we start from all the visitors need to start from algae strain room, and then algae scale up, and the larval section, and maturation section, and breeding program wing C and wing G, and commercial multiplier wings, wing D, E, F, and then finally to the water systems. And all the tools and equipments we separated from section wise. Actually, we have different tools, and commercial multiplier wings we have, and commercial multiplier raceways we have different tools. In NBC, we are following 5-step water treatment process. In step 1, we are doing mechanical filters by sand and gravel. And step 2, we are disinfecting water by ozone. And again, in step 3, again, we are filtering water with 1 micron cartridge, and then carbon filters, and then finally UV. Each step, we have different validations for sand and for plankton validation. We are collecting plankton from the backwash and filters, and we are validating for WSSV. And for after ozone and after UV, we are validating for microbiology for total pet count, total Vibrio, and total green Vibrio. We are pumping water from Red Sea, and we are passing the water to mechanical filters. We are using sand and gravel as a filter media. Sand, we are using 60% in the filters. Sand size is 1 to 2 mm size, and gravel we are using 40% and 3 to 4 mm size. The water for total, for maximum retention of total suspended solids and plankton, we are using sequence and sand filters. We have 10 sand filters, 2 in the seaside and 8 sand filters inside the facility. And in the next stage, water we are disinfecting with ozone. Ozone is a very strong oxidation agent with oxidation reduction potential of 2.07 compared to chlorine and the chlorine dioxide. Ozone is a powerful agent. And ozone is very unstable gas. We cannot be stored, so we are using ozone generators, and we are injecting the ozone into the ozone towers. The water coming from after sand filters, the water coming into the ozone towers, and we are injecting the ozone into the oxygen towers. And when the ozone mixes with water, it oxidizes and start eliminating all the virus and bacteria. We are using ozone above 650 millivolts. After disinfection, the water we are storing in the water reservoirs. We have total 3 reservoirs. Each reservoir is around 500 cubic capacity. And from the reservoirs, we are pumping the water to 1 micron cartridge filters. From 1 micron, again we are pumping water to carbon filters, and finally water is going to the UV and then UV chambers. These are the microbiology validation reports for year-wise data from 2017 to 2022. All the batch we produce, we are validating for microbiology. Once we get the results, 0, and then we are pumping water to the production facility. We are collecting daily plankton for WSSV from backwash sand filter. From NBC lift station, effluent treatment pond, and also from pump station. So far we did 2,500 samples, and we got 13 positives, and all the positives are from the seaside sand filters. In NBC we are using squid and polychaete in only maturation section for broodstock conditioning. We are purchasing fresh fish from Topsy Bites from Thailand, from Holland, and GB Aqua, squid from GB Aqua, USA. These are the PCR reports from the supplier. And once the shipment we receive to NAQUA, again we are validating for WSSV, IHHNV, EMS, and microsporidia. These are the PCR reports for this year's shipments. And all the other results are negative. And these are the yearly data of fresh feed validation from 2018 to 2022. We are validating for WSSV, IHNV, EMS, and microsporidia. And all the results were negative. We are validating our broodstock health by PCR, histology, and wet mount analysis in all the stages of broodstock. First, we are validating the parents for IHHNV and histology, and once we get the Nauplia negative, we are transferring the Nauplia to larval section. In larval section, we are sending the samples for cold challenge and analysis and testing for all the OA listed pathogens: WSSV, BP, TSV, IHHNV, EMS, EHP, YHP, IMNV, CMNV, NHP, MBV, and HPV and microsporidia. And in the juvenile and broodstock stage, we are collecting all the daily mortalities for WSSV, and SAS sampling is visiting every 3 months and collecting the samples during this stage from juvenile and broodstock stage in NBC. And also we are keeping our broodstock in the inland genetic backup facility. They are collecting the samples from both NBC and inland genetic backup facility, and then analyzing the samples for WSSV, BP, TSV, IHHNV, EMS, EHP, and NHP. And also we are validating when we transfer broodstock for commercial purpose to commercial hatcheries. We are again validating for histology and WSSV. These are the yearly data for broodstock screening for PCR from 2013 to 2022. We did around 65,000 samples. Most of them were for WSSV and BP. All the results are negative. And the Brewster screening by histology from 2013 to 2022, we did around 7,500 animals for histology. All the results were negative. And Brewster screening for PCR for all the import batches, we received from 2013 to 2014, we received 13 shipments from Ecuador, and 2018 to 2021, we received 6 shipments from Thailand. All the shipments we did first quarantine and then send the samples for University of Arizona and Jeddah Fish Health and Safety Laboratory and NACWA Diagnostic Laboratory for WSSV, IHHNV, IMNV, NSV, TSV and YHV, and all the results were negative. And Brewster screening for PCR in histology by MEWA for WSSV, IHHNV, TSV, NHV, YHV, EMS and EHP. From 2016 to 2022, they did samples for 2,000 animals for PCR and 700 animals for histology. All the results were negative. These are the latest MEWA PCR report from samples from NBC. And all the results were negative. And also they are taking samples from the inland genetic backup facility, and all the results are negative. These are the MEWA's SPF certificates, this 2021 certificate and 2022. And then MEWA is certifying for free of WSSV, TSV, YHV, NHP, EHP, IMNV, IHHNV, EMS and DIV. In 2016, September 20, NACWA and CP Group Thailand, we have a memorandum of understandings to exchange our genetics. NACWA provided our WSSV tolerant animals to Thailand, and we received fast growth line animals from CP. As a part of collaboration, we sent 6 shipments to Thailand, and all our animals are tested in CP Diagnostic Lab and Department of History's Thailand Diagnostic Lab. And this is a PCR report from Thailand for our animals tested for WSSV, IHHNV, YHV, TSV, IMNV, IHHNV, and AHPND. All the results were negative, and also they did PCR for in Department of History's Diagnostic Lab, and all the results are negative. In 2019, July 15th, Department of History's people came for audit, and they checked all our quarantine reports, biosecurity surveillance reports and national surveillance reports, and they audited and they give the certificates. In 2019, we got a score of 96%, and 2021 we got a score of 93%, and in 2022 we got a score of 91%. We got low score because we are not doing brute stock sale. That's the reason why in the last 2 years we got a very low score. Thank you.
Welcome back, ladies and gentlemen. We are going to start again. Now I would like to invite our next presenter, Mr. Mustafa, and he is going to present regarding shrimp diseases in KSA and abroad. Please, Mustafa.
Okay, good morning everyone again. So I will be talking about the major shrimp diseases that is present in KSA, and especially in KSA and abroad as well. So as we all aware that this shrimp industry has a rapid growth for past few decades, and even though it's growing very fast, but still we are facing some problem with especially significant loss due to infectious and non-infectious diseases. And infectious diseases, it's mainly like viral pathogens, and especially WSSV, it can cause as a serious challenge in this shrimp industry, and especially even in the KSA. The main objective of this presentation to provide the understanding about the shrimp diseases, what are the causative agents, clinical signs, and what are the precautions to be taken to avoid any recurrence. Okay, these are the major shrimp diseases listed by World Organization of Animal Health and MIWA based on the relevance to Saudi Arabia. So this is the World Organization of Animal Health listed diseases. This is the KSA listed diseases, and it is the pathogens detected in KSA. So here you can see these are all the pathogens detected in KSA. And apart from this, we included this emerging disease, that is EHP, that all we know is that it does not make any mortalities, major mortalities, but it can make huge impact in the production because of low growth. It will stunt the growth, retard the growth. But this is still, it is not yet listed in World Organization of Animal Health. So we will go for one by one. White spot syndrome virus. The speciality of this virus, this is the only virus within this family, and this is the biggest genome in the earth. Okay, so even though if you are considering the humans and animals, this is the biggest genome. That is the speciality for white spot, and this is the rod-shaped double-stranded DNA virus. So if each diseases we will go for this causative agent, behavioral signs, what is the susceptible species, and it is reported, mortality, transmission, what is the diagnostic method, target organ for the test, preventive measures. Okay, so in white spot case, the behavioral sign is, we know anoxia, it's lethargy, reddish colorations, and especially the white spot in carapace and other exoskeleton parts. But it is, recently it is very easy to identify if we have any white spot. It is easy to identify within carapace. We can see white spot, and especially in exoskeleton parts as well. But in recently, for example, in Vanami, in our case, if we have any white spot outbreak, the spot is not visible. Even we can see some animals, we can see like in the reddish color, but sometimes even though not reddish color, even though there is no white spot, still we are detecting positives. So sometimes we are detecting some white spot in the carapace and in the exoskeleton parts, but it could be some other bacterial infection as well. So the susceptible species, we know that most crustaceans and including crabs, and it is first reported in Taiwan in 1992. But it doesn't mean that it is the origin of disease in Taiwan, because Taiwan they have more high research facilities. That is the reason they detected in first. So after the Taiwan, you can see the China, Japan, Korea, Thailand, and other countries. The mortality, it is starting after 2 to 3 days of infection, around 80 to 90%, and it's continuously 5 to 7 days onset of mortalities. But nowadays, if you are using SPF and SPT stock, the mortality it's reduced. And for example, if you have any drop of temperature or other environmental factors, it can cause of mortality, but not like 80 to 90%. The transmission through vertical and horizontal. Vertical means it's inside the eggs, and water, soil, crustaceans, planktons, these are the transmissions. So diagnostic method, PCR histopathology and immunohistochemistry. The target organ for testing, all ages of postlarvae, gills and pleopods. Preventive measures, SPT, SPF, UV, ozonized water in hatcheries, proper die-out, and biosecurity measures. Tora syndrome virus. This is a RNA virus, single-stranded RNA virus, causative agents. The behavioral sign is it is especially in appearing in pre-molt stages, and it is appearing red color, and particularly in the tail region. You can see especially in the tail region, it can appear in the red color. It is susceptible for widespread, including the Vanami, Monodon, and Indicus. It is first reported in Ecuador in 1992, and it is spread all over the Latin America, and including in Saudi Arabia, it is reported in TSP. And the mortality observation is especially during the molting stage, 5 to 95%, and especially it is occurring in the juveniles, and adult is more resistant. It is usually occur in more in juvenile stage. The transmission, it is vertical, it's not inside the eggs, it is around the eggs, and horizontal transmission. The diagnostic method, PCR and histopathology, target organs, larvae, gills, muscles and pleopods. The preventive measures, SPF stock, UV and ozonized water, avoid introduction of wild broodstock, and strict biosecurity measures. IHHNV, the main causative agent is it's caused by single-stranded DNA virus. It's containing still parvovirus. What is the behavioral sign is, before Peter mentioned, especially we can see its deformities and rent, and it can reduce the feeding and elevate the mortality rates. It is susceptible for several venom species, and including especially it is most susceptible for this Stilrosteris, and it is all the life stages of Venami. And it is first reported in America in 1981, but they are mentioning that they suggested that the source it was from the Philippines, and it is also reported in Saudi Arabia. The mortality, IHHNV does not cause high mortality, it is lethal infections, and the transmission it is through the vertical and horizontal. Diagnostic method, PCR and histopathology. Target organ is larvae, priopod and gills. Preventive measures, SPF stock, disinfection of eggs and nuclei, and proper dry out, strict biosecurity measures. So for IHHNV, PCR it is very sensitive because it is detects viable and non-viable pathogens, as Dr. Victoria mentioned before, because sometimes it can give a false positive due to the presence of EVs, means endogenous virus elements. So World Organization of Animal Health, it's adapted the methodology to it's not adapted the methodology to differentiate EVs from the infections. So there is no scientific evidence for the pathogenic with aquatic products, and it does not cause severe mortality. And there is an effort to remove from this list. EHP we mentioned that this is other emerging pathogens, but it does not cause any mortality, and it is very highly resistant and stay longer in environment without any host. So it is present in terrestrial, freshwater and marine ecosystems. Okay. This is actually this causative agent is obligatory intracellular spore forming fungi. Okay. The behavioral sign is slow growth, significant size variations. It is susceptible for monodon, Venami and Stilrosteris, and it's first reported in Thailand 2009, and it is other Asian countries including Saudi Arabia reported, and but it is not listed in Hua. Mortality, it does not cause any significant mortality. It is retard the shrimp growth, and it is severe economic losses in shrimp farms, and it increases susceptibility of bacterial infections. So it can cause some other secondary infection. Transmission through vertical and horizontal transmission, the main root cause is oral and fecal contaminations. So the diagnostic method, PCR is the most suitable method, and histopathology and even wet mount. But out of this diagnostic method, PCR is the most suitable method, and histopathology is very difficult to visualize, even wet mount. So the target organ for the test, hepatopancreas, and all the stages of size of post larvae. The preventive measures, maturation in hatchery. For example, in the hatchery case, for the maturation feeding, we need to freeze up to -20 degrees, and for the processed food, we need to cook up to more than 75 degrees Celsius. And we need to use UV and ozonated water and EHP free live feeds, for example, in Artemia case. For ponds, we need to go for proper dry out and disinfection between the cycles, and we need to ensure that EHP spores along the cycles, and we need to destroy them. For the surfaces, for example, in the tank, we need to use the oxidizing like 65% active chlorine or 75% of ethanol or KMNO4.
Mustafa, just one question. Is that the only one, EHP is the only one which goes to the processed food side or to the consumption side as well? Or other ones, the other viruses also carry on for the food?
No, for
if
the,
they are not treated well?
no, if they are treating, it is not going to the other foods.
No, but that's the only one where you mentioned 75 degrees centigrade processed food?
This is for especially for the spores, to destroy the spores.
The spores are more resistant than the higher particles.
It is more resistant.
Not to introduce when you are feeding the broodstock, so you inactivate it before you feed. So there is no infection process because it can also be
Yes, but
through
he's saying that
Processed
it
foods
can be
carry on being
other
spores.
viruses.
Being spores, won't they be carry on if they are with the shrimps?
We freeze them. They want the fishes,
Freeze
we
minus
freeze
20.
them. Minus 20. All the fresh feeds in hydration goes to freezing.
so it inactivates.
Inactive.
There is no
Inactive.
Inactive.
It
The
must be
spore
infection.
best is the freezing.
Yeah.
And the second thing is retard the shrimp growth.
Yes.
What does that mean?
Retard means the shrimp it will not grow. It will stop the growth. Yes, in the small stage. It will not grow.
Irregular or small? Just small.
Even after the stocking, yes, it will retard the growth. It will not grow. Even though our FCR will grow up, but the shrimp will not grow. Yes.
Economically, it has terrible impact.
For example, in maturation, for example, in NPU, nuclear stages, we are using for the broodstock. What we mentioned that freezing. For example, we are feeding squid or artemia. We need to freeze up to minus 20 degrees. Then it will inactivate the
So
spores.
in the picture here on the side, you have very small and you have very big. So is that irregular or retard?
Yeah, this, the main thing is behavioral, the science is two things. It is slow growth, size variation, and it will retard the growth as well. Because, for example, if we stock in one pond, we have a big population. Maybe the EHP is infected in a specific population,
And
not
kept some
all.
small,
Yes.
but the other one will
Yes. So you can see the
Depends on the level of infection.
infections.
If it's more than 10 to the 3 per microliter, then it has very high impact. If it's below 10 to the 3, it is no impact in growth. So it depends very much on the level of infection of each individual.
Yeah, it is explaining for the coming slides. Yeah. So this is the diagnostics by wet mount for EHP. We just include this slide because here is this is the protocol. Place a drop of fluoxetine B-stein, distilled water in a microscopic slide, and remove the tissue from hepatopancreas and to the specimen, and put in the staining solutions, and remove the largest pieces, cover slip, and observe slightly egg-shaped spores. You can see here each is spores in hepatopancreas. Here with smear, see the small spores. Here also in wet mount and EHP from the fishes, but it is very difficult to identify by using the wet mount. For the EHP, what's the impact and management? It is highly linked to crop intensity. It is affect the growth, as mentioned earlier. If it is less than 10⁻³ copies, it will not make any negative effect. Okay, not make any mortality. If it is more than 10⁻⁷ copies, it can cause chronic mortality. Okay, so it can cause some other disease also. So it is for other secondary infections. And for example, white species syndrome, the combination of EHP plus infection with other anaerobic bacteria. Okay. To way forward this exclusion in broodstock in larvae, we need to clean broodstock, pH oscillation, density, we need to stock below density 30 V per meter square and maintain the high oxygen level. So here is the examples. There is a successful harvest in Thailand in 2022 with Mr. Robin McIntosh. They did not use any chlorine. There is any healthy other fungi chemicals. They have laterite bottom. The aeration is 350 kilograms per HP. And what this only difference is there is 5 ppt salinity. There is only small ponds, 1,500 meters square. They use paddle wheels, and then stocking rate is 120 per meter square. So they have made successful harvest even though there is a EHP positive. So BP, this the causative agent is Baculovirus pinea. The behavioral sign is less feed intake and increase the gills and surface fouling. It is susceptible for all the species. It is first reported in Gulf of Mexico and Central North America, Hawaii and KSA. The mortality is especially in larval and post-larval stages, and 100% within 3 to 4 days. But at present, it will not cause any high mortality, even though we detected BP in aqua before, but we eliminated successfully. The transmission, it is through vertical and outside the eggs. Vertical means it's vertical and horizontal, but vertical it is outside the eggs. The main important things for BP, it's a quality control criteria in the hatchery. Especially what we need to do, we need to wash the eggs properly to eliminate, because this is outside the eggs. So if you are washing the eggs properly, we can eliminate it from the NPU itself. Okay. So there is another method is anal sealing before spawning to eradicate BP from the broodstock. EMS or AHPND, the main causality, it is not, actually this is not infection, but it is toxicosis. So it's a bacterial origin with the strain of Vibrio parahemolyticus. The behavioral sign is reduced feed intake and pale discoloration of hepatopancreas, you can see in the pics. It is susceptible to Venami, Monodon and Chinesis. It is first reported in China and then other Asian countries. Initially it was reported as early mortality syndrome because of mass mortality, it is happening from the early stages. Okay. So mass mortality, it is happening within 35 days of grow-out culture. Transmission is vertical and horizontal. Diagnostic method, PCR and histopathology. This is the target organs, post-larval and hepatopancreas. The preventive measures is SPF broodstock, test in larvae before stocking, strict biosecurity rule. The main thing is, for the preventive measures, we need to reduce the substrate, especially in nursery and grow-out stages, that feed for bacteria. So we need to remove the excess sediments, excess foods, and maintain the low level of bacterial replication. So, for example, if you want to test the EMS, first we need to increase the sensitivity of testing. So, if we are looking for EMS in the feed, for example, looking into the water, first we need to incubate the feed into the mesh, put a little bit feed in the mesh, and we need to incubate in the tank or in the pool for 6 hours and extract the DNA from the pellet. Okay. That's if you are looking for the water. So if you are looking to the animals in the grow out, for example, we need to remove the animals outside the water, and we need to go for self incubation minimum 4 hours, and we will go for the DNA extraction. So in breeders, we need to can take a feces and incubate TSB agar 2% for 4 hours 30 degrees. In larvae it's the same, we can incubate 4 hours 30 degrees. So then we will go for the testing. So these are the method to increase the sensitivity of testing. So as we discussed before, from the experts, Mr. David Kawaishi and Mr. Robert McIntosh, they are also suggesting to management strategy to roll out EMS or control EMS to prevent or a prevention. First of all, we need to keep the pond bottom clean. The priority number 1. The priority number 2, maintain the clean bottom. Priority 3, make sure the pond bottoms are clean. Okay, yes. So the same, that means we need to remove the dead algae, organic waste, molds or uneaten feeds. We need to make the pond bottom clean. So the feed, excess feed for the bacteria, they cannot able to colonize. Okay. So yellowhead virus, the causative agent is it's a single-stranded. This is RNA virus. This behavioral sign is stop feeding suddenly at one point, and it's light yellowish color in hepatopancreas. But usually the people are saying yellowhead virus. You can see the hepatopancreas color is yellow, but it can it's not because of these diseases, but there is some other, other reasons behind this yellow. Okay, basically we can see the stop feeding suddenly and swimming near to the ponds. That means we can see very weak animals. It's first reported in Thailand and other Asian countries, and it is highly prevalent in the population from Australia. So the mortality from juvenile stage, 5 to 15 grams, it is up to 100%. Transmission vertical and horizontal. Vertical means it is inside the eggs. Diagnostic method, PCR and histopathology. Target organs is all stages of post-larvae and gills. Preventive measures, SPF broodstock, proper disinfection in between the cycles, and strict biosecurity measures. HMNV, infectious myonecrosis virus. This is also RNA virus. The behavioral sign is whitish necrotic area, especially in the tail. You can see whitish necrosis area, and it's a little bit reddish color. It is susceptible for Venami and other species. It is first reported in Brazil and other countries. Mortality is its range to 40 to 70%, and FCR it will go very higher, and it cause very big economic loss in Brazil. 2002 to 2006 period. Transmission vertical and horizontal, it is outside the eggs. So diagnostic method, PCR and histopathology. Target organ is gill, striated muscles, and all post larvae. Preventive measures, disinfection, proper biosecurity measures, proper dry out. NHP, the causative agents, it's Rickettsia-like bacterium. The behavioral sign is, hepatopancreas become like watery. You can see this, just compare this, become watery within our black sticks, and high FCR. That means high FCR means the animals is not growing. So it is retarded the growth as well. It is susceptible for Venami, Monodon, and Chinesis. It is first reported in China and the other countries. It is mortality 20 to 90% within 30 days of appear in the clinical signs. But at present, it is not cause any big mortality. Diagnostic method, PCR and histopathology. Target organ, hepatopancreas, fecal matter, different post larvae, different sizes. Preventive measures, SPF broodstock, pond dry out, especially in between the cycles. Early detection also, it's very important. MPV, monodon baculovirus. This is a single double-stranded DNA virus. This the behavioral sign is dark colors. You can see the animals look like dark colors, and acute. It's loss of hepatopancreatic tubules. So it's consequently disinfection these organs. So lately hepatopancreas, it will go for a disinfection. So it is susceptible for indicus and monodon. First reported in China and other countries. It is mortality. It is increased the chance of secondary infection because of poor growth and mortalities. Transmission, it is vertical and horizontal outside the eggs. Same like same like TSV, this is outside the eggs. So the quality control in hatchery is very important. So if you are using properly washing the eggs, so we can rule out this from the hatchery. So diagnostic method, PCR and histopathology. Target organ is all stages of post larvae and hepatopancreas. Proper dry out, strict biosecurity rules. So this is SHIV or decapod iridescent virus, as Chakravarti mentioned before. It's iridescent virus. It's a double-stranded DNA genome. It's a large ecosystral shape. Its behavioral sign is pale or yellowish discolorations in hepatopancreas, and slightly, of course, we can see the weak shrimp show slightly discoloration. It's one of the behavioral signs. It is susceptible for these all species, including crayfish and all life stages. First reported in China, and later on it is reported to Thailand at low prevalence, but it's not officially confirmed. Mortality 14 to 16%, and 18% reported overall. But there is no massive mortality reported. Transmission, vertical and horizontal. Diagnostic, PCR and histopathology. Target organs, gills, hepatopancreas, lymphoid organs, rostrum. Preventive measures, screening of broodstock, virus-free fresh feeds, and water treatment of UV and ozonation. is HPV, hepatopancreatic parvovirus. This is a single-stranded DNA virus. This behavioral sign is atrophy of hepatopancreas, anorexia and slow growth, and of course gill fouling. It is susceptible for all these species. It is reported in Singapore in 1984. It's marine farmed fish. Mortality, no massive mortality reported. Transmission through vertical and horizontal. Transmission diagnostic method, PCR and histopathology. All stages of post larvae, hepatopancreas is the target organ for the test. Strict biosecurity and reducing the pond density, that is the preventive measures. White fishes syndrome, it is unclear. There are many causative agents behind this. Still it is not clear. So it is associated, it is associated with Gregarian worms, vibriosis or EHP, or it is aggregated transformed microvilli structures in hepatotubules. So there are many findings, but it's still not clear. Behavioral sign is like whitish fecal strings. You can see whitish fecal strings in the water, and it is very less active, sluggish swimming. These are the behavioral signs. It is susceptible to vanami and monodon. It is first reported in Thailand, later on India. It is affected, it's like 17% of shrimp farms. Mortality, it is within 10 days, it is floating on the surface in the period of 10 to 45 days after the stocking. It is FCR will be, it is elevating the FCR, of course the growth reduction and size variations. Transmissions, vertical and horizontal, but vertical it's not clear still. Diagnostic method, PCR and histopathology. Target organ, gut, hepatopancreas and all the stages. Preventive measures, proper dry out in between the cycles, better management practice, biosecurity measures, treatment of salt pond sediments by Q-Clim 6 ton per hectare, that has been recommended. Cotton shrimp diseases, CSD, the causative agent is Microsporidia. Behavioral sign is white opaque muscle, slightly infected, and heavily you can see this white opaque appearance. Okay, so these are the behavioral signs. It is susceptible for many shrimp species, such as including the crustacean, lobsters and crabs. It is reported in Madagascar and Mozambique, Asia and Latin America. Its mortality, it is low level of mortality. Transmission through horizontal transmission, from transmission disease from shrimp, fish and shrimp. So there is an intermediate host in between fish, shrimp, fish and shrimp. Okay, the diagnostic method, PCR and histopathology and wet mount. The target organ, muscle gills and hepatopancreas and all the stages in larvae. The preventive measures, we can exclude the intermediate host, for example for the fish. If you remove the fish in our population, in our from the ponds, we can you can prevent this cotton shrimp disease, and proper dye out and disinfection is recommended. So these are the references. Thank you so much. I'm sorry because it was very fast, but we don't have much time, and it's good I present after the lunch.
Thank you very much, Mustafa, for giving a very good overview of diseases in KSA, shrimp diseases in KSA and abroad. Now I would like to invite Mr. Shoaib T. Muhammad for the similar overview of diseases of fish within KSA and abroad.
Thank you, Dr. Tariq. Assalamu alaikum and good morning. So, I will quickly jump to the list of diseases because Mustafa already gave the introduction. There are okay. Sorry. Okay. These are the list of diseases, especially listed by OE and MIWA based on relevance to KSA. As you can see here, there are 5 viral diseases, 2 bacterial and 1 fungus and 1 parasite. And this is the status of these diseases in KSA. We have red sea bream iridovirus, RSIV, BN9, big belly syndrome, and streptococcus is already present in Saudi Arabia. And there are 2 emerging diseases which is not yet listed. They are scale drop disease and it's a virus disease, and tilapia lake virus. And this is economically important based on KSA experience. I think it is very relevant for us, which is brucellinosis, it's a parasitic disease, Thanasibacillum, which is bacteria, and amylodinium and Sphaerocotyl chrysophory, both are parasitic diseases. And let's move on to VNN, viral nervous necrosis. The status is endemic, it's present in Saudi Arabia, it's caused by nodavirus, and these are the different genotypes of nodavirus. There are several kinds, and the behavioral sign is they show abnormal swimming behavior. They will go whirling swimming, sometimes upside down, and in flat fishes they don't show much clinical signs. They will stay in the bottom, but they will be like bended. Okay, and gross pathology is hemorrhagic brain, and they are susceptible for over 50 species, and which includes European sea bass, gilded sea bream, barramundi, groupers, and everything. And it is reported in South and East Asia, many of the countries, and Australia, Mediterranean, all this, France, Greece, and all these countries, and also in Saudi Arabia. I have marked this in red because we have introduced VNN through imports. We have introduced European sea bass from Cyprus, which was infected. 2-3 shipments was positive. Okay, and it is also found in our Red Sea. The mortality ranges from 80 to 100%. Early stages it dies faster, and if they are older fish, it is 50 to 60%, which is also depends on the species. In our experience, as we got VNN, the mortalities were much lower in older fishes. And transmission is through vertical and or horizontal. And diagnostic method, we have PCR and histopathology, and also immunohistochemistry. The target organ for this virus is brain, spinal cord, and retina. And the preventive measures, it is very important to use SPF broodstock. So we don't have any more vertical transmission. And we cannot use farm raised or wild broodstock because we do not know their health status. It can be carriers, as Dr. Victoria was telling. They may not show the clinical signs when they are bigger, but they are carriers. They can pass the infection to the larvae, and it can cause serious problem. So, and also use of water treatment, UV ozonation for hatchery operations, and avoid feeding trash fish, because even if you have the best broodstock without any problems, if you can infect them by giving contaminated feeds. And also do vaccination. There is no treatment available. This is how it looks like in histology. There will be a lot of vacuolization in brain. And iridovirus, RSIV, it is also endemic to Saudi Arabia. We have seen some positives in wild. It was groupers, and it is caused by RSIV, is a DNA virus. The behavioral signs is they get lethargic, and they also show some visible respiratory movements. And susceptible over 40 species, which includes pompano, amberjack, sea breams, groupers, barramundi, all the fishes that of our interest also. And the susceptible age is juvenile to adult, but juveniles are more susceptible. They can cause high mortality. And risk factors is always, usually with summer season, when the temperature is about 25 degrees. It's reported in Japan, Taiwan, China, Hong Kong, and also in Saudi Arabia. When we talk about this behavioral science, they are mostly non-specific. We cannot just conclude from that, because all the diseases that I am going to explain, most of them will have similar kind of clinical signs. So it just gives you a clue that you need to go for further analysis, as Peter was explaining. And mortality can go very high in early stages, even up to 100%. If they are bigger, there will be lower mortalities. And transmission is through contaminated water, can involve some vertical transmission, is not yet proven, but we still recommend always use SPF broodstock, because we don't know what will be passing to the larvae. And always use water treatment and vaccination. And diagnostic methods, we have histology, PCR, and we can do organ imprint of spleen. It will be easy to identify, and hematology there will be low hematocrit value. And target organ is spleen, kidney, heart, liver, intestine, and gill. This is how it looks like in histology.
So this is only for the sequel?
No, it can be.
It's called as sequel.
Yes, it can be over 40 species, but the name is.
Since it was identified firstly from Red Sea fish.
Yeah, so they put it.
That's what they named.
And there is so many.
Cannibalism is there, so maybe all of the species can eat one and.
This can be found in different species. Yes, if they are eaten, but
it can be found in another species as well.
Yeah.
Not only the sequel.
Yeah. And let's move to scale drop disease. Very important for barramundi. It is also a viral disease. It's also iridovirus, same as the one before. It's iridovirus, DNA virus, and the more behavioral and morphological signs, they will have abnormal nervous behavior, swimming behavior, and there will be spiral swimming, which I already told you for VNN. This is non-specific, and there will be a lot of scale loss. One of my colleague who Eric Wiss from he was working in Singapore. He said this is one of the worst thing he ever see. If you dive into a cage and the animal looks weak, you try to hold them, they just poof, and your hand is full of scales. Yeah. Susceptible species, it is only reported in barramundi as of now, and it was found reported first in Malaysia, and it is in Singapore and many other Southeast Asian countries. Cumulative mortality can be 40 to 50%, and it can go both juvenile and adult fish, and it seems to follow a seasonal pattern with their experience. It's like inter-monsoon or around September. It's related to monsoon. Yeah. And transmission is horizontal. We don't know if there is vertical. There is no confirmation, but still we have to emphasize on using SPF broodstock. And diagnostic method, there is PCR, there is histology. Is all the scary photos you can see. There can be lesions in liver, kidney, then brain and every other organs. So, and there is no treatment, and preventive measure is isolate the affected population to avoid spread, and terminate the stock or do an emergency harvest, and disinfect the area, and remove dead and dying fish to, the weak fish or dead and dying fish is like a factory of pathogens. So you just remove them regularly. And use of SPF broodstock, very, very important, and avoid using farm raised or wild broodstock. Just imagine if we are going to a cage which was infected with scale drop, which we don't know, and you take animal and bring it to your hatchery, you amplify them. Yeah. Okay, it's very important use of SPF. Water treatment is important, and general rules of biosecurity, disinfection and everything. And next is tilapia-like virus, again viral disease. Its behavioral and macroscopic signs are lethargy and abnormal swimming at surface. There will be no schooling. They will be lonely, isolated, and swirling and loss of balance, loss of appetite, and there will, this is important, ocular lesions and skin lesions. And susceptible species is wild tilapia, farm tilapia, hybrid tilapia, or this tilapia, and it is reported in Ecuador, Colombia, Egypt, Israel, and Thailand, India, everywhere. And affected stages, different people have different opinion. Some of them got even yolk sac larvae, early stages. When they archive the sample and check for PCR, they turn positive. But in Israel, it was a wide range of sizes were affected. In Egypt, more than 100 gram animals were dying, and they thought it is like a summer disease. But later on, they checked for TALB, and it was positive. So it affects many sizes. And mortality can kill approximately 90% of population in fingerlings within a month of stocking into cages. At bigger stages, it causes 9% mortality. Risk factors, they say it is always associated with transfer between ponds and cages. Is handling, stress and all, it will be additional stress you give, so they express more disease and cause mortality. Transmission is horizontal by cohabitation, and vertical transmission is suspected because we already see positives in yolk sac larvae and all. And diagnostic method is PCR, and histology, again a lot of lesions in brain, eye, ruptured and cataract changes, and liver, then also in spleen. And next is streptococcus is very important also. It is present in Saudi Arabia and it used to cause a lot of problems before in barramundi and also in tilapia. And it's caused by Streptococcus ineae and Streptococcus agalactiae, they are gram-positive bacteria. And behavioral signs, again it is also having swirling movements, pop eyes, you see here, hemorrhagic eyes, hemorrhagic lesions, and loss of appetite and anorexia, and they get thin. And gross pathology is hemorrhage internal organs. You dissect and open, you will see bleeding there also, hemorrhagic lesions, and pale livers and enlarged spleen. Susceptible species, over 27 marine and freshwater fish species, which includes barramundi, rainbow trout and yellow tail. Reported in Asia, Europe, Japan, US, it's also in Saudi Arabia. Mortality can kill up to 80% depending on the age and intensity of infection, and transmission is vertical outside ovum and also horizontal. Diagnostic method is organ imprinting. It is very rapid, faster, cheaper, and microbiology, PCR and histology, and target organs are liver, spleen, kidney, brain and bloodstream. Preventive measures, of course, always use SPF broodstock, avoid farm raised or wild broodstock, and do water treatment for hatcheries. And there is vaccination, so we have done vaccination and it is very successful for our barramundi. And treatment, in case of mortality, we can go for antibiotic treatment by oxytetracycline. Big belly syndrome, also very important. It causes mass mortality in early stages, especially in nursery stages. It is caused by intracellular bacteria, Vibrio-like species like bacteria, and the behavioral signs is bloated belly, knife like tail. It will big belly and very thin tail. As you can see here, it's like very thin tail. And there will be discoloration, they will be isolated, no schooling behavior, and the affected range is 0.5 to 5 gram. When they are bigger, they don't show much, but in earlier stages they can cause much problems. And there is also detected in 200 gram fish, but with very, very low mortality. And it is found in Saudi Arabia, it's endemic to Saudi Arabia, and it is reported in Southeast Asia, Australia, which is not official yet, and also in Saudi Arabia. And mortality, it can cause as bad as 80% mortality. It usually starts after a handling, like grading or movement and all. And transmission is through contaminated feed and cannibalism. Preventive measure is again use of SPF broodstock, avoid farm or wild broodstock, water treatment, and maintain early stages at low salinity. This bacteria will not multiply when they are at low salinity. So the best thing you can do, this early stages, nursery stages, you rear them in low salinity. So there will be no multiplication. Once they are above 5 gram, there is no problem. They don't cause any problems. I have some video, but we don't have time for that. And diagnostic method is rapid staining, organ imprinting, very easy, as Peter was saying, within an hour you have the result, and very cheap, PCR and histology, and maintain salinity at low temperature, and antibiotic fluoramphenicol is useful to treat them. Next is Thanasibaclosis caused by Thanasibacillum maritimum, Thanasibacillum dicendraki, and there are different other species. And the clinical signs is skin lesions and yellowish patches in gills and eroded mouth and all of them. And susceptible species, it's widely distributed. Many species are getting infected with Thanasibacillum. In our industries, Paramondi, bream, sea bass, starboard, everything will be infected. And stress factor, they require a primary cause. It can be handling, lesions caused by handling, grading or something. Then the Thanasibacillum will infect them.
Just a quick question.
Yeah.
These infections,
Yeah.
are they local or systemic to the, for example, if there is a 2 kilogram fish,
Yeah.
and you have a sign of that?
It can attack any stages, juvenile to bigger.
Yeah,
Even
but
if
within one animal, one big animal,
Yeah.
if you see a lesion like this and it has infection showing in one area of the fish,
Yeah.
does it go to the all?
it can
flesh?
also go. It can even get inside.
Okay,
Yeah.
so it's
Usually
systemic.
they are outside, but in certain cases they even get inside. Rare, but usually outside, in skin, in gills.
But this one normally
It's very rare they get inside, very rare.
So
wherever there is a skin
if you
lesion.
remove that, you can still.
Yes, you treat them.
The problem is when they do back when they do transplant, so they damage the skin and then you get a secondary infection. That's
They get
This one
exposed.
is local, right? This one's local.
They get whenever there is a damage, they get exposed and they can get it. And you see in sole and turbot, this is how it looks like in gills, yellowish patches. Yeah. And transmission is horizontal. Mortality can be significant based on the level of damage. And diagnostic method is fresh wet mount. You just take and look under microscope and you will see this hair-like, mesh-like bacteria. And preventive measure is minimize handling stress and vaccination. Treatment can be done by a peroxide bath, freshwater bath, antibiotic treatment, or OTC in case of increased mortality. And next is mycobacteriosis. It's also a bacterial disease caused by a variety of species of mycobacterium. And behavioral signs, they can also go thin, they can have exophthalmic eyes, which is pop eyes, loss of appetite, and in case of broodstock, they can also affect reproductive problems. And you see here lesions here, and very important, they can have a lot of granulomas, nodules in internal organs, gray or white nodules in internal organs. Susceptible species, broad range, freshwater, marine and brackish water environments, all those species can be infected. Transmission is vertical and horizontal, and it's very clear in live bearing fishes, vertical transmission. Diagnostic method by PCR and histology, and target organ is especially is spleen, kidney and liver, and there is no treatment, no approved treatment. And preventive measures is like do a quarantine and depopulation, euthanasia and depopulation, and disinfection protocols, very important to get rid of all those bacteria. And this is how it looks like inside, all these white granulomas. And it's very important, it is zoonotic. So those who handle fish, they always should use proper protection, gloves and all, because you can get infected and you can have all these problems. It's also called, that's why it's also called fish tank granuloma or fish handler's disease. And next is pasteurellosis, again another bacterial infection caused by Photobacterium pesticida, Photobacterium Damsella pesticida, and it's also known as pseudotuberculosis and or photobacteriosis. Behavioral signs again, loss of weight and darkening and necrosis in gills. Non-specific also all the signs. And if you dissect and open, you will see internal organs with white nodules. That is why it's called pseudo tuberculosis. And susceptible species is wide variety of species, marine, including yellowtail, sea bream, sea bass, cobia, pompano, and all. It was reported first in the USA, and it is in Europe, Asia, North America, everywhere, and Japan, Taiwan. And this is how it looks like. You see the little white nodules in spleen, and this enlarged spleen. And they also have a pale or greenish fluid inside. A mortality can go from 90 to 100% in early stages, and once they are bigger, they are strong, so the mortalities are lower. And transmission is mainly through horizontal mechanisms and via fecal or oral route of transmission. And diagnostic method, bacteriology and PCR, and they can infect liver, kidney, or spleen. That's the target organs. And the preventive measures is to avoid all kind of stress, overcrowding, and use of SPF broodstock, avoid farm-raised or wild broodstock, of course water treatment, and prophylactic chemotherapy, sulfonamides, nitrofurans, and all this antimicrobial we can use as prophylactic. And there is vaccination and antibiotic treatment if it is needed. That's all about few of the bacterial diseases. There are plenty more, so I was just choosing some. And next, go to fungal diseases. It is epizootic ulcerative syndrome, or EUS. It can be in freshwater, estuarine, and marine. And it is also called red spot disease or mycotic granulomatous and ulcerative mycosis. The causative organism is fungus, Aphanomyces invadens, and Aphanomyces pesticida. And behavioral signs: lethargy, reduced appetite, erratic swimming, and so on. And clinic, it can have localized swelling. It can have small pinpoint red spots like hemorrhagic spots on body, and localized raised areas with hemorrhages, and protruded scales and scale loss in some cases. And it is firstly reported in Japan and later on Australia and India, Philippines, and is in many countries also. And in gross pathology, ulcer can be found broad, but at the center with necrotic, highly necrotic area. It can infect over 50 species, both farmed and wild fish, and freshwater and estuarine. And bottom delving fishes like morels, like snakehead, and catfishes are more susceptible. Target organ is skeletal muscle.
Can you make it quick because...
Sorry. Transmission is horizontal. Mortality can go over 50%. Diagnostic method, histology is very important. We can do wet mount, but histology is very important. Control measures, selection of resistant species is very important, and we can do treatment for using Cifax, slime, and K104. And parasitic disease, this can be a nightmare. Bruclinella, it can affect especially sea bream. Just imagine a cage with this much of mortality. They kill just like that. It's a ciliate. It can it can be treated rapidly with low dosage of formalin, but they go very fast and they jump between cages so fast. They show clinical signs: respiratory distress, hypersecretion of mucus, and it is present in Saudi Arabia. We got very bad hit before, and also in Mediterranean regions. Transmission is through contaminated water, and very important thing, it has some kind of seasonal pattern. When the temperature is high, we used to get it. So the precautionary measure is increase surveillance during the summer period. That is most likely to happen. So increase surveillance and ensure you have the formalin stock. You find it. If you don't have formalin, you take 2 more days. It can be disaster. And prophylactic formalin bath based on your observation. and wet mount, histology, and we can do for diagnosis. Trichotinosis is also a parasitic disease caused by Trichodinella. It's very common, it's widely worldwide present, and mortality can go up to 50% in case of severe infections. In our case, it is very low mortality. We didn't have much of problems because we immediately act, and it didn't cause much mortality. It can have marine environment and also found in fresh water. Transmission is through contaminated water, diagnosed with fresh wet mount and histology. Prophylaxis, of course, avoid stress, avoid more organic loads in the tanks, and high water exchange treatment. 50 ppm of formalin is very effective, and hypersalinity, malachite green, potassium permanganate, and fish will recover immediately after the treatment. Amyloidinium is very important also, and tricky one. It is caused by Amyloidinium ocellatus. It is a parasitic dinoflagellate. The behavioral sign is, shortness of breath, jerky and flashing and swimming at the surface. And susceptible species is so many species, and sea bream and sea bass and turbot and all part of it reported worldwide, and it is also in marine and brackish water. Mortality can go even up to 100% depending on your culture condition. If you are in a area without more exchange like in lagoon, it can cause much higher mortality. This is how it looks like in gills. No time for video. And the life cycle of this parasite is very, very important, because they get infect the animal in the gills, and they grow there, and it is called trophon. They grow and they detach, they go to the pond bottom or tank bottom, and they will develop from there, and it will hatch out a lot of dinospores. Dinospores, which is the infectious form. So it's the whole cycle. So the problem is, your animals get infected. If you are inside a tank, all these tophons will go down, and they will come back and reinfect you. And these tophons are resistant to the treatment. All you can kill is these dinospores. So you need to have extended treatment. It's not one day. Sometimes it needs to go 2 weeks of formalin bath to make sure all the generations are killed. And it's contaminated water, which we already explained. Prophylaxis, maintaining good fish health condition, filtration, disinfection for hatchery, UV and quarantine. And this is also need to be considered. Frequent removal of dead and weak animals, and displacement of cage location. If it is in cage, if too much there, because we know it goes down and it can come back. So move the cage location. Or if it is in tank, you get all the animals and move to other to avoid reoccurring. Next is Paracotyle chrysophory. It's a monogenean parasite, susceptible species sea bass, sea bream, and other sparids. It's also reported widely in many countries, and low mortality in viscous fish. Transmission is through contaminated water. Diagnostic method is fresh wet mount, gill scraping, and histology. Treatment is formalin bath or hydrogen peroxide bath. Cryptocarrion irritants, or called marine white spot disease, is caused by ciliated protozoan parasite, cryptocarrion irritants, and similar, flashy and distress in swim abnormally. That is shortness of breath. That is the behavioral signs, and they used to have small nodules in the body and also in gills, as you see here. Even if it is not there, sometimes it will not show in some species if they are pale. So you will have to look under microscope to confirm. Target organ is gills and skin. Transmission is horizontal. Control measures, quarantine, use UV and frequent bottom cleaning, displacement of cage if possible, change tank every 3 days. Again, this also have they can reinfect. And treatment, copper sulfate, formalin, and hyposalinity, reduced salinity. Next is amoeba gill disease, caused by amoeba, Neoparamoeba purens, and they can also cause gray or white mucoid swellings in gills, and they will have impairment in gas exchange and problems. It can affect respiration, osmoregulation, and and there's susceptible species is salmon, and many other species is susceptible. Target organ is gills. You see, this is how it looks like in gills. So there will be no exchange or excretion through gills. Reported in many countries. You can detect by wet mount or histology, higher mortality in adult fish, and preventive measure is do bath, freshwater bath for 2-3 hours up to 12 to 15 times, and treatment again freshwater bath, copper sulfate, chlorine dioxide, all those chemicals can be used to treat. Enterospora nucleophila, it is an internal parasite. Sorry, it's caused by microsporidia, and behavioral science. They get very thin, and their intestine will be very thin, and they will affect the growth, and sometimes white feces. And mortality can be it's low level, but it will affect the growth. Diagnostic method: wet mount, PCR, and histopathology. Target organ is stomach. Treatment: no treatment. Enteromyxosis is another parasite, metazoan parasite. It also affect the gut and stomach, and again the animal will look like very thin, emaciation. And susceptible species is white giant, sea bream, sea bass, turbot also included. It is reported in Mediterranean in 1980s, and it is also, as per literature, it is also present in Red Sea. Mortality is temperature dependent, and in various species mortality is different. It can be high mortality in turbot and sheephead bream. Transmission is horizontal and vertical also. Preventive measure, improving husbandry strategies basically, and also avoid using the mass fruit stock, and also UV and ozone treatment, and periodical epidemiological surveys for early detection of disease. And treatment, they cannot survive in freshwater, so hyposalinity is a good way to treat them. Thank you. Sorry I took more time. It was a big topic.
Thank you very much. Actually, it requires a lot of time to understand all these things, but thank you very much again. As per increasing aquaculture production, so there are more exports and imports from various countries. So all the countries have their own regulations, like KSA has implemented various regulations for production, biosecurity, import and export. To understand the scientist status and the market access, I would like to invite Mr. Shabir Alam Muhammad for the presentation, please. Mr. Shabir, thank you.
Bismillahirrahmanirrahim. Assalamu alaikum rahmatullah. Thank you for today. We had a very detailed technical presentation, and all the SPF products and all shrimp production or fish production. The purpose is to take to the market, and how successfully market the products is very important, very essential for commercial reasons. So let us start. We all know that SPF status, which is the basic and absence of specific pathogens from a population of shrimp or fish. And the Saudi Arabia Vision 2030 requires aquaculture establishments to produce 600,000 tons of shrimp and fish products. It's very important. So for a sustainable shrimp and fish production, healthy broodstock and PLs are mandatory. So we will go to the legal basis of KSA. What are the legal regulations in Saudi Arabia, which is focused on the aquaculture production and various stages of. First is the National Aquaculture Policies and Practices, which talks about the production system, registrations, approvals, use of drugs, antibiotics and all. The second one is the National Biosecurity Manual for the shrimp and fish culture. There are two manuals are there for the biosecurity. It is the most important manuals in the biosecurity risk mitigation. And the National Aquaculture Import Manual focused on the imported shrimp and fish species, especially on the importation of the live animals, and what are the regulations to be followed. The National Aquaculture Products Export Manual just talk about the export requirement. If you are exporting any products, shrimp and fish products, frozen products, or fresh products, or live products, what are the requirements to be met? So the summary of that, focusing on today's topic, switching to SPF stocks is primary requirement under the National Biosecurity Program. And the importation of SPF stocks permissible by MIWA when no stocks available in KSA. And necessary biosecurity protocols shall be applied by aquaculture establishments during importation process. So this is the basic things. So why Saudi Arabia offer SPF shrimp? So I'm focusing on the shrimp side because the 80%, 90% of our export is based on the shrimps. So to achieve the disease-free status of the shrimp production, and many countries, importing countries, they mandate import requirements to have the shrimps from free of diseases. For example, if you go to the Australia, there one of the health certification requirement, and one of the approval requirement of the country or health certification requirement, they ask for a product from each batch has been found processing to be free of white spot disease or yellow head virus based on a sampling and testing methods recognized by the OIE for demonstrating the absence of disease. You can see that they are asking, Australia is asking each batch to be free of disease. It's very, very particular. At the same time, China, they clearly specify that it should be products that were inspected and quarantined by competent authority, that is in Saudi Arabia, MIWA, and not found by any pathogenic organisms, harmful substances, and foreign substances. This is related to food safety in PR, and the diseases listed by OIE. They clearly specify that all products which are exported to the China should be free of diseases which is listed by OIE. Today we have already seen which are the OIE listed diseases for the shrimp and fish in different presentations. And at the same time, for each China shipment, it is very important to submit a test report from NFC, National Fisheries and JFRC lab, that our shipment is free of diseases. The same, going to the South Korea, they have similar requirement. Even South Korea, same like Australia, they specified all the list of viruses which is need to be free from their shipments. Okay, and also we need to provide the each shipment virus-free report from the KSA labs. Thailand, which is an emerging country, which we need to export in future, which is also talking about the free from all OE listed diseases. So in the importation manuals and the import manual, which we have already mentioned, which referred previously, which are the approved species in Saudi Arabia by the Ministry of Agriculture. These are the 9 fish species, approved species, which SPF species we can import, and 1 shrimp species approved for the importing live for the SPF purpose. Which are the SPF breeding facilities? Today we have already seen that NACWA nuclear breeding center is already approved breeding facility for the shrimp. At the same time, for the fish species, since we don't have the SPF breeding facility in Saudi Arabia, we are importing most of the SPF fish from different countries following the National Aquaculture Import Manual requirements. So how to import the SPF shrimp or PLs? There is a very simple process, but there is step-by-step process controlled by covering the different risk of the importation and all. First thing is to apply with the MIWA with the approved species, listed approved species. Any new species we want to import, that has to be agreed by the MIWA. MIWA approved and the species to be imported from the MIWA approved hatcheries. They will provide you with the list of approved hatcheries, which has to be, you can, if you want any aquaculture establishment, they want to import SPF shrimp or SPF fish, PLs or broodstock, we have to follow the MIWA approved aquaculture facilities from the out of KSA or within KSA. Import permit it's mandatory because we need to go through the custom clearance. Custom clearance, they specifically require the import permit. Any new species, as I told, this has to be reported MIWA. MIWA will conduct the import risk analysis. What is the risk of Saudi Arabia if we import these species? And they also approve the hatchery and SPF facility after the physical inspection with the industry collaboration with the SAS, and they complete the first cycle. Second cycle, at the same time, commercial establishments, they should ensure and all the proper documentation for the importation is completed. Some of the examples I have given here. The commercial invoice, which is packing list, what type of packing you are doing, what are the quantities you are packing, what is it contains, water how much is contained, live fish PL, how many numbers, all these details should be provided. Health certificate, which is issued by the different exporting countries, like if you are importing from Australia, any Baramundi, so that the health certificate which is issued by the Australian authorities also important to clear the shipment in the Jeddah port or any other ports. Virus-free report, which is coming, this is not the virus-free report, this is the second stage. But the first stage virus-free report should come from the Australian authorities. Then the second stage virus-free report should, once we receive from when it reaches to the quarantine, JFRC labs, they collect the samples and they issue the virus-free report here. The quarantine facility, the commercial establishment, they should coordinate with the respective MIWA agencies to ensure that all imported stocks are going to a quarantine facility, or aquaculture establishment, they should have a sufficient quarantine zone for storing the imported stocks separated from their own stocks. It's very, very important. And the quarantine period to be declared by the Ministry of Agriculture, MIWA. So MIWA samples testing, as I told, that MIWA with SAS, they collect the samples from the seaport or airport, which are the locations, and or also they will visit quarantine facility if required. Export of SPF shrimp. Right now, Saudi Arabia, they have only one species which is which is a shrimp. And they have potential to export to various destinations as the market demands. So the export of establishment is very, very easy. They have to apply with the health certificate of the MIWA branches and virus-free report of the fish health and safety labs, and other documentation as usual. So the overall nutshell of this our today's topic, the recommendation is the aquaculture farms and hatcheries shall fully ensure using SPF shrimp and fish products in the production system. It is basic. When our foundation is strong, we use then we can provide the final product to the countries strongly without any hassle. The final product reaches to the different countries like China and other countries. They found any of our products with any listed diseases, they will simply hold the containers either to destroy or to bring it back. Most of the times they destroy. It's very difficult. So whatever we do effort in the farms, hatcheries, processing, we don't follow the biosecurity protocols. It is going to affect when the product reaches to the destinations. So it is highly important today, whatever the topics we covered, that has to be implemented. Also, to develop SPF NBC facility for the key commercial species in KSA. This is also important. Thank you very much. Any questions?
So, just to be clear, so every country have certificates for the country. Before we deal with the for Australia, South Korea, Thailand, and China, right?
For the export. For the export.
Yeah. So export. So what about European Union?
European Union also have similar, but they don't have stringent requirement. They don't have strict requirement because in European Union, shrimp culture is much lesser than these countries. These countries, they have the more shrimp culture.
Yeah.
European Union, they don't have much shrimp culture.
So that's why
Yeah, that's why. That's why.
One more thing, Shabir, I think you rightly mentioned in the recommendation part that in case of anything positive, definitely they will hold the shipments. But for KSA, if something happens to one exporter, everybody in KSA will be affected with that. So everybody have to pitch in their part, small, big. It's not about only just one company. It's about entire the nation can be affected by if we get anything, any infractions in our export protocols.
Basically, it is not one establishment is approved. For different countries, they have different regulations. Like some countries, they approve a specific establishment, but some countries they are approving the whole country.
So what about the certificates here required? It's the same like our rules is different. Do you have any?
Yes. We different countries have different rules.
Yeah.
The basic rule they follow is that disease is free from the OE list. Yeah, the basic rule. If we have Saudi Arabia, as you told on the presentation, our objective is to completely control the risk of all OE diseases plus additional some known diseases. So once we have free, then only we can strongly export. So when we are doing this SPF programs, we also have to consider these requirements of other countries where we are going to export. What we are doing SPF not only for us, this is for commercial reasons. So we are at the same time we are meeting the different country requirements.
And what about import?
I would like to add here actually, all countries have health certificate requirements. Before the issuance of certificate or sending the certificate to the regional branches to issue, actually there is a G2G certificate consultation, health certificate consultation. So, like let's say we are exporting to Australia. They send us the requirements. The competent authority, MIWA, receives the requirements. Okay, we gave them the assurance that we can fulfill all the requirements, and the health certificate is finalized with the approval of both the competent authorities. Once it is finalized, we distribute it to the regional branches also, because the regional branches are issuing the health certificates along with the head office.
Yeah.
So for export, for import, okay, if we are exporting, import also.
You have like stickers.
Yes, we have import requirements. KSA also has import requirements.
But for the import, KSA import, it is managed by Saudi FDA in coordination with the MIWA. Yeah, import of frozen products, live products by MIWA. Yeah, live product directly it will go to the MIWA. For the frozen products, by import approvals done by Saudi FDA in coordination with the MIWA. Yeah.
So import or
Yeah.
collaboration with
For the import, all the establishment they have to register with Saudi FDA. Any establishment they want to, like Dubai, if they want to import any frozen products, they have to register with the Saudi FDA. Then take the approval before the export.
Any other question, please? That's it? Okay.
Thank you.
So, here we reach to an end of this workshop. I would like to thank everybody who spared their time and participated in this workshop. Hopefully we all got benefited by the presentations. Thank you very much, NACWA team, all the establishments, National Compliance, Environmental Compliance Center and NIWA people. Now, before leaving, we are going to have a group photo, and after that we are moving towards Flamingo Mall. At first floor of Flamingo Mall, there is Barbecue Tonight restaurant. We will have an open buffet there. So the third floor? Okay. So people who are having their own conveyance, okay, they may reach there, or the one who are not having, they can accompany us. We have transportation facility, like so after group photo we will wait for the people who are going with us. Tomorrow, yes please. Tomorrow we are going to NACWA for SQ facility visit as well as NBC visit.
started with
Everybody who is using their own car, they should reach there by 9 o'clock or before 9 o'clock. And anybody who want to accompany us, we will arrange the transportation from here So you should be here right before 7 o'clock. At 7 o'clock we will reach from here. That is fine. 7 o'clock we will reach from here. So any person who wants to go with us, you are welcome. We will be here waiting for you. And any person who wants to go using their own vehicle, you are also welcome to go.
So it's better that people who are coming from Jeddah, they reach there at one time,
yes,
so that you get inside the gates at one time, instead of having one person coming half
So
an hour.
hopefully
very hard to manage.
We are leaving from here at 7 o'clock and we will be there at 9 o'clock. It's 2 hours now.
So that everybody gathers at the gate.
Up to the gate. Is there or if you present another 45, 50 people to go to the facility?
That will be achieved.
Please come in casuals.
We welcome you guys at the gate.
You all will come in casuals.
अस्सलामु अलैकुम औज़ु बिल्लाहि मिनश शैतानिर रजीम बिस्मिल्लाहिर रहमानिर रहीम अल्हम्दुलिल्लाहि रब्बिल आलमीन वस्सलातु वस्सलामु अला रसूलिल करीम सय्यिदिना मुहम्मद व अला आलिहि व सहबिहि अजमईन. First of all, from the board of directors of NAQA, from the management team and all the members of NAQA family, I welcome you here at NAQA Palace and at the NAQA project. Basically, it's a history which was written about 40 some years ago, 1982-83, started with an R&D project, research and development project, and from there, with the help of Kingdom of Saudi Arabia and with the help of the native, hardworking, dedicated nationals, citizens of Saudi Arabia, this project has come to be one of the only 2 or 3 few top line totally integrated aquaculture projects in the world, and has become a benchmark for the fishery industries, fish, shrimp and other related products industries in the world. So, I will go through, I will just take maybe total of 10 minutes of your time, and then we watch a 3 or 4 minutes of corporate video, and if there is any question you have, Mr. Shabir will answer that. Okay. So, welcome to the MEWA, welcome to the NCEC, SAS and all the friends who they have come from aquaculture industry. This is a snapshot of NAQA's 70 kilometers coastline, and in this coastline we also have about 1100 square kilometers of sea projects which we have our certification for, but about 250 square kilometers of inland production of shrimp and fish and other products happening here. And as you know that this is about 182 or 180 kilometers away from Jeddah, depending upon where you are. Somebody measure it from Makkah Kubri, some measure it from Bariman and some from Asfan. So, depending upon where you are, it goes from somewhere from 170 to 200 kilometers. So, as you can see, this is the island where we have all our hatcheries and our LRU productions, and I think the NBC is also on the same island, which is a natural barrier from its all production areas and sea, and then we have lagoons in front and then our projects. This is we call it headquarter or head office. We also have a corporate office in Jeddah, which is our business office, but this one is our operational headquarter. This is our senior management team or executive team. Mr. Diego is our CEO, he is from Ecuador, he is coming from a fisheries background. Then you see here Mr. Arfin is the Chief Human Resources Officer, Amir Khan is the Chief Financial Officer, this is myself, I am the Chief Quality and Sustainability Officer. We have Director of Shrimp BU Business Unit, Eric Wiz is the Director of Fish Farming, Engineer Alawi Hussain is the Director of Feed Processing and our Utility Services. You have met with Dr. Victoria, she is the Director of Biosecurity, very close to what work you guys do. Dr. Khalid Al-Balla is our Director of our MENA Sales. Shivalan Ko is the Director of International Sales, and our newly acquired Mr. Akram Fawad is the Director of Supply Chain for NACWA. So, this is the executive team. NACWA, like I said, about 40 years ago started in 1982 as a research and development project, which then moved into an experimental stage. From there it went to commercial operations in 1995, 2001, and industrial operations started in 2001 to 2010, mean we start building the processing plants in this time. And the fifth stage was diversification of our projects. We bought a lot of allied products in there. Sea cucumber started, we had algae productions here, so many other projects. We started the phase 2 of NACWA with it. So, this is where it started growing. Now, sixth stage, what is sixth stage for us? Our next stage is basically to comply with, to help and to be committed to Kingdom's Vision 2030. So, as you guys know that Kingdom of Saudi Arabia has made a commitment of having the abundant, feasible, approachable, economical food supply to the nationals of Saudi Arabia, and to increase its exports into the aquaculture products. That is one of the part of the 2030 vision, where I think MEWA and SAS are basically fighting on the front lines for us, and then all of the industry and the allied forces that we have, we are helping towards that. So, NACWA is also part of that game. So, our production, our capabilities, our manpower, our intellectual properties will grow and increase as the production of that, I think 600,000 tons, 600,000 ton is the target of having the fisheries production within Saudi Arabia. NACWA is committed to very soon hit the 100,000 tons mark and then go beyond, inshallah, in that side. At this time in MENA, in entire Middle East and North Africa, we are about 85% of the production of the fishery products. NACWA is already holding it. We have 4,000 employees, 32 countries, people are coming from all over the world. We speak about 19 languages when we are nice, and we are angry, maybe 32 languages. We have 550 Saudi nationals and maybe more, inshallah, and we also have sisters. This is good. I joined NACWA in my first term when in 2013 to 2018. At that time we had, I think, about 30 or 40 ladies working in NACWA in a very small section, but alhamdulillah right now closer to 200 women are part of NACWA family. So, this is very encouraging, and it is really along with the vision of Saudi Arabia. Here is our processing plant. No, this is our power plant, sorry. This is power plant, as we said that we are integrated system. So, we produce our own water, we have our own desalination plants, we have our own RO plants, we have our own power, we generate our own transportation, we are all the way from start to the end totally integrated project. So, about 43 megawatt of power is being produced by NACWA inside. 120,000 tons of feed mill, this is part of our feed mill. I think you guys have not visited the entire project. So, that is why that was the purpose of giving you this little presentation to introduce NACWA to you, because you might have seen only one area of the system. So, this is our own feed mill, our processing plant. This is one of the largest shrimp manufacturing plant in the world, which has the capacity of 1,000 tons per day, and alhamdulillah, I think in 2018 or 2019 we have already touched that, right? 2018, yes. So, our own hatcheries and nurseries, then you can see these areas of nurseries, and you might have also seen some of these sites. These are our grow outs. So, NACWA basically on the fisheries side we can produce about 10,000 tons of fish, mainly barramundi, which is sea bass. That is our main production of the fish. We also have tilapia, some other species that we grow, but mainly barramundi at this time. So, about 3,000 tons of tilapia per year about 10,000 tons of barramundi. That's our production at this moment, but we have more licenses and we can produce more if we need to. On the shrimp side, we have about 500 ponds of 10 hectares each. The total production, like I said, NACWA is targeting 60,000 tons, closer to 60,000 tons this year inshallah, and next years to come we want to increase our production to 100,000 ton, and we have the capability and on ground facilities already for that. We can go to that level, that's no issues. So 100,000 tons of fishery products are on the way to NACWA. Our water management, our intakes from sea through the main feeder canals, then going through all our ponds about 5,000 hectares, and from there it goes back through our environmental channels with all the controls of environmental and in compliance with the national NCEC. We have people from NCEC here or not? We have? No. We don't have? Okay. So anyway, we are in compliance with all the water which is being used going through our main drainage canals, then going to elite mixing zones, and this is where our controls are, not controls but also our sampling points are. Controls are done in multiple layers ahead of it, and everywhere we are taking samples and checking all our parameters. So about 7,000 parameters are being tested in NACWA every year for environmental only. Sales distribution to 32 countries. This is our barramundi. You can see tilapia, red sea tilapia as well, part of our product line. Then sea cucumber, that is also our production. You have seen some of the pictures outside. These are different species that we are working with KAUST to develop more. We have a world level quality control system, quality assurance system, which we call NACWA IMS, Integrated Management System of Quality, which has analytical services, which has regulatory services like Mr. Shabir works on compliance and regulatory department, he heads that one. Satish is here for aquaculture quality. We have Vilas who works and heads the supply chain quality side, and then we have Mr. Ismail who is the head of the Central Analytical Services. We have other departments as well like SQA, we have shelf life studies going on, so many things happening in the quality sector side. And on the biosecurity, you guys know much better, NACWA has a world class biosecurity system and being benchmarked all over the world, okay, recognized and also well controlled and monitored in the leadership of Dr. Victoria. These are different certifications that we have. We recently have acquired ASC, which is the Aquaculture Stewardship Council, and one of the stringent and most strict environmental and foods. Yes. So it's and also with MSc, COC, chain of custody. We also have BAP, we have GAP, we have Saudi GAP and global GAP as well. We used to have BRC as well, and we are still in compliance with that. For some reason we have dropped the certification. ISO, all kind of ISO certification, HACCP, GMP, FDA compliance, EU compliance, SASO compliance, NCEC compliance. Any more compliance? Yes. So the wildlife compliance. So alhamdulillah, I think more than about 20 different type of global certification and approval system is already established in NACWA. These are some of our products. You will see them in the market, and especially this is our new products coming up soon. You will see skin wraps and individual packings. This is pride of our place, late King Abdullah, may Allah bless his soul. This palace was basically built at that time when he came in and he stayed at NACWA project itself. So it gives us a lot of pride and pleasure that we are supported and we are blessed by their efforts on the back. Sheikh Al Raji you can see in this picture as well. These are different prince and they are coming and visiting Nakwa. Here you can see Prince Khalid Al Faisal. He was the governor and he is the governor of Makkah, I think, even still. Yeah. Okay, this is something which I think Shabir can relate to, but this is Alhamdulillah in 2015 we became the world only first company which had 4-star BAP for Baramundi. So we not only we produced more Baramundi than Australia. Australia was the one that they gave us the brood stock to start Baramundi in Saudi Arabia, and then Nakwa became much more producer of Baramundi than Australia itself, and even still that goal is with us, that target is with us, Alhamdulillah. So at that time when we became the 4-star BAP best aquaculture practices 4-star certificate holder, our minister went to Boston to receive the award, and then when they came back, King issued this letter to Nakwa, and this is something which really gave us a boost to say, if we are working for the kingdom, if we are working for the vision of the kingdom, then Alhamdulillah the people who they lead this kingdom are also recognizing it. They gave us encouragement to do more. Okay, so that is something which we are really proud of, Alhamdulillah. Okay, we have a small. Thank you very much. Okay, this one is a small video. I hope that it will be self-explanatory. Can we turn the lights off?
180 kilometers south of Jeddah, surrounded by desert, you will find the National Aquaculture Group on the shores of the richest, most saline waters in the world. With 200 square kilometers of land and 65 kilometers of coastline, National Aquaculture Group is designed to produce more than 100,000 tons a year of the very finest quality seafood. Clear about our objectives from the outset, the company started building one of the largest vertically integrated farms in the world. In this remote part of the world, everything had to be built from the ground up, including basic support facilities like providing water and electricity. A 42 megawatt power plant and hundreds of kilometers of roads and canals are part of the essentials to develop this 200 square kilometer farm. 3 pumping stations with a capacity of 150 cubic meters per second lift fresh, pristine seawater to the major canal 2.5 meters above. The water flows down naturally through a network of canals to provide the shrimps with fresh water in the 4,000 hectares of ponds. For the 2,500 people living here, all life's necessities such as schools, laundry, supermarket and entertainment centres have been provided. Producing our own feed for the shrimp and fish is essential to ensuring the quality of our products and a key element in vertical integration. Our feed mill produces more than 10 tons per hour of diets formulated from the highest quality raw materials. Maintaining the highest levels of biosecurity is critical for our industry. Animal health is vitally important. The feed and water quality are controlled on a daily basis in our state-of-the-art laboratories. Highly qualified technicians ensure that all national aquaculture products meet the highest international standards for both food safety and environmental sustainability. Red algae, or Dunaliella salina, is grown in large ponds using the rich fertile Red Sea water. These algae are an excellent source of natural carotenoids for both animal and human nutrition and natural food products. Our most biosecure area is the breeding centre, where we keep our brood stock. The shrimp hatchery is producing more than 5 billion Norpli and more than 2 billion post-larvae a year. Offshore, we currently have 3 sea cage farms in operation. Each has 20 cages, and each farm has the capacity to produce 5,000 metric tons of fish annually. National Aquaculture Group grows shrimp in more than 500 inland ponds, each with an average size of 10 hectares in a semi-closed system. Our annual production capacity will grow to 35,000 metric tons of shrimp, and we aim to harvest 9 months of every year. After harvesting, the shrimp is transported in temperature-controlled containers to our processing plant. Even shrimp from the most distant of our farms will be packed and frozen in less than 7 hours. Using the very latest technology, the fish processing plant can process between 40 and 80 tons a day, depending on the type of end product required. The shrimp processing plant can process more than 150 tons of shrimp per day in 2 shifts. Several freezing methods are available, like semi-individually quick frozen, block frozen, in brine, individually quick frozen, and even sushi quality grade produce. We use cutting-edge software for yield control and traceability, ensuring we remain at the forefront of our industry. At National Aquaculture Group, we take pride in making a difference. National Aquaculture Group. A vision to a new horizon.
We have Vaikam here. He is one of the great marine scientists right here. Satish is here, the aquaculture quality champion. Shabir from regulatory side. Shokat has also worked as the HOD of our analytical services for NAQUA. We are so proud of him. So any question, guys? Okay, you guys are most welcome.
We are happy to hear.
Mashallah.
I hope when we go back, we will get some feedback.
When you go back? Yeah, in the stores there are a lot. No, I'm just kidding. No, we should have some gifts sometime.
NAQUA is setting so many benchmarks, achieving so many milestones, and now the NAQUA is like an institution, like an academy for learning.
Yeah.
Is there any plan in future, like keeping in view Vision 2030, like institutes, like other institutions in the world, like NAQUA Academy for Aquaculture courses or something like institution for learning? So
Right.
some kind of this, is there any plan in future?
Actually, NAQUA already have started a general aquaculture quality aquaculture academy, and in combination with the government as well. So we have some collaboration going on with them already. NAQUA actually, before COVID, NAQUA had a full plan of even building a center of excellence for NAQUA, but I think that as the profitability and producibility of NAQUA will increase, we will go to that side. And having a third party institute attached with it will be the next stage, inshallah. It's a good idea. We should think about it. But actually you see that so many people who I see around have been part of NAQUA journey and have learned and have taught us in so many ways.
Yes.
Right? Amina, you were also, you got some training from here, right? Yeah. So it's two-way. Basically, when somebody comes from outside and they get trained at NAQUA, we also learn from them. So it's an open subject. Okay, guys? Huh?
It's not a university.
Allah Kareem. We are still students. We are learning.
It's like a university, complete.
Yeah.
System is there.
Vaikam, what you say? They said they build a university here. I mean, me, myself, I have a degree in food science and technology. I am not a marine scientist. I'm not a DVM, a veterinary medicine. But I learned, So this is the beauty of NAQUA, that we have people from so many backgrounds. We have chemists working here. We have, microbiologists, biologists working here. We have marine scientists working here. We all put together our knowledge, and this is how an FMCG company works, a global level company. It doesn't just take one side and the way look at one thing. I worked for Nestle, Pepsi in my background, and those experiences helped me here to produce the fishery products for the consumer. So it's not just a farm, it's producing the fish and fishery products for 32 countries of the world and consumers. So converting a farm product into a table product, into a consumer product, this is where we all help each other and our backgrounds will help. Okay. Khas, thank you very much, guys. Thanks a lot. You guys are most welcome again, but I think it's time to go. So, Doctor, you are the leader of the team. What you say?
And it was a great learning, not only even the workshop, but also here at the NBC. Your staff is, mashallah, really competent. From A to Z, they have explained things very well, and surely it needs a lot of time to understand everything, but still, we got many things from here. Thank you very much for everything.
Let's give a hand
Thank
to
you.
each other and have a group photo here.
Yeah.
Come on, guys.